Hey everyone, welcome to the Drive Podcast. I'm your host, Peter Attia. Rhonda, it is so great to see you, especially on that awesome remote setup that I almost shouldn't have said to people was remote, because it really feels like we are in person, about as close to in person as we've been in a few years.
It's really great. It's really great to see you, Peter. It's been a minute.
Yeah. Well, we'll meet together soon in a couple of months, so I'm looking forward to that. There's a lot to catch up on. Over the past few years, we've obviously exchanged a bunch of emails about things that we each find interesting. And I think in the last couple of years, well, let's just pause it, in the last 5 years was the last time we did a podcast. Just going back to that point, I think both of us have evolved a lot in our thinking. And I think we've done so unapologetically. That's the nature of science. That's the nature of what we do and we're trying to learn. So in thinking about our discussion today, I think we both agreed it would be most enjoyable to at least spend some time talking about areas where your thinking has evolved. But I think first we wanted to start with, I don't want to put words in your mouth, but maybe that which you're thinking about the most right now. Would that be a safe assessment if we were to start to talk about dementia, specifically Alzheimer's disease, and maybe the change in how you think about that?
It would. For me personally, I have neurodegenerative disease on my mind quite a lot because Alzheimer's disease and Parkinson's disease both run in my family. So, and I have a genetic predisposition. So for me, understanding everything I can do, with my diet, with my lifestyle, just, exposure to or limiting exposure to certain things, et cetera, becomes paramount, right? Because I don't want to get Alzheimer's disease and Parkinson's disease. So, as you know, Peter, the Alzheimer's disease field it's been quite a roller coaster in a way. We've had this dominating sort of, I think, hypothesis, this amyloid hypothesis, as it's called. So there's, one of the major pathologies of Alzheimer's disease are amyloid plaques in the brain. And there are other pathologies, tau tangles, also glucose hypometabolism. So, glucose uptake into the brain is impaired, and also perhaps even the utilization of glucose as well. So these are like three major pathologies of Alzheimer's disease. And it seems as though the majority of targeting, how science and scientists have decided to target, Alzheimer's disease is through this anti-amyloid, hypothesis. And as you know, we've had quite a few failed trials, although of recent, a little bit more, I would say, possible success maybe. But generally speaking, it's like we need to go, okay, are we just trying to treat a symptom here, or are we too far downstream? Like what's the deal? And I started reading some studies by Borisov Slovovic at USC and Dr. Axel Montaigne, who was trained with Dr. Slovovic and now has his own lab at the University of Edinburgh in Scotland. And I recently did a podcast with him on my podcast, and Axel. And it was really like, when I started to read some of this literature, and I'm going to talk about like what, this sort of new, it's not even necessarily new, but like, it's not a new way of understanding it, but it is in a way, because in the public opinion, in the public mind, it's a new way. And this is sort of like, okay, well, what are the underlying causes of dementia? And so there's, three major types of dementia, Alzheimer's disease being like the most common. There's small vessel disease, cerebral small vessel disease, and then vascular dementia. Those are the three most common forms of dementia. But like, is there a common underlying denominator between those? And on top of that, like what sort of lifestyle factors and genetic factors do we know really increase the risk of Alzheimer's disease and dementia? Well, we know having an ApoE4 allele, so this is a version of a gene that is known to increase the risk of Alzheimer's disease. If you have one of them, it increases the risk twofold. If you have two, if you got one from mom and one from dad, it could be up to tenfold. And this isn't like an early onset Alzheimer's disease. It's more of what's called late onset, which is the normal sort of age-related, progression of Alzheimer's disease. But that gene really does play a role in someone's risk. The other thing we know is type 2 diabetes, right? That over, somewhere between 50 to 80% of people with Alzheimer's disease also have type 2 diabetes. Like, that's a lot. There's definitely something going on, right?
Yeah.
And this is where you hear.
And let's sort of pause there for a moment, because I want to go back to sort of the premise of your interest, which is you are at increased risk. So even if you just think about this personally, and therefore presumably you're interested in quote unquote prevention. And as you've probably heard on my podcast, prevention is still a word that doesn't quite resonate within the field, which is, in other words, up until very recently, I think the NIH didn't even acknowledge the idea of prevention as a strategy within this field. And in fact, preventative neurology, or preventive neurology, I suppose, is really something that is still kind of on the outskirts. Most people are thinking about what to do when you have Alzheimer's disease. Not as many people are thinking about the question that you're asking and that a few other people are asking, which is, what's in our control? Because yes, you alluded to ApoE4, which I'm sure we'll talk more about, but what you just said about type 2 diabetes would suggest that if you believe you can prevent type 2 diabetes, wouldn't that at least suggest you have the probability or possibility of preventing or delaying Alzheimer's disease? Sorry, so I didn't mean to interrupt you, but I want to highlight the important implication of that statement.
Exactly. It's so important. I think that looking at Alzheimer's disease and understanding sort of the underlying cause of it opens up, these new avenues for prevention and also treatment, and of course there are people that do get Alzheimer's disease early in life. These are people that could come down with it in their 40s or 50s. That's sort of people are outliers in that case, but it does happen. And so obviously there are things that you might do everything right and still have that terrible genetic combination, right? So but with respect to the ApoE4 and type 2 diabetes, understanding again, is there something common going on here that we can sort of understand as a foundation to what are the initial things going wrong to lead to Alzheimer's disease? And that is where vascular dysfunction, particularly the blood vessels and capillaries that are lining the blood-brain barrier, seems to be a really, really early event that is common between all types of dementia and between type 2 diabetes and ApoE4. So people with type 2 diabetes, as you know...
Can I interrupt for a sec, yeah. Rhonda? Tell folks what the blood-brain barrier is. I think it's obviously going to be an important part of this discussion, and not everybody might understand what it is or why it's so important.
So the blood-brain barrier, and this is, again, I am not a neuroscientist. I'm just, I am a scientist, but I have an interest in this. And so I've done a lot of reading in this field. The blood-brain barrier serves a couple of functions. One is it is providing, there's a combination of different cell types that make up the blood-brain barrier and a lot of vasculature, right? And so blood flow, things are brought to the blood-brain barrier, and oxygen, glucose, other nutrients, and they are, transported across the blood-brain barrier. But it also, as the word implies, provides a barrier to things that you don't want to get into your brain. So, we don't want red blood cells getting into our brain. We don't want a variety of other molecules, proteins that are floating around in our circulation to get into our brain. And so when the blood-brain barrier begins to break down, and it's kind of people I think are a lot more familiar with when the gut barrier starts to break down. I think it's a sort of become more of a common theme that people are focused on gut health. And so you'll hear the word, mostly you hear the word leaky gut. I don't really like that term. I think it's intestinal permeability, but the tight junctions, these proteins that are holding endothelial cells together in the gut, when those open up, you get that term, leaky gut, or as it really should be called, intestinal permeability. Well, in the brain, you also have endothelial cells and you have tight junctions. And when those tight junctions also break apart, and we can talk about like what's, at the root of that, that leads to permeability of the blood-brain barrier. And therefore, two things happen. One, you are allowing then things from circulation to get in the brain, which wreaks havoc on the brain and sort of leads to this vicious cycle of neuroinflammation, inflammation in the brain. But also, you're disrupting the transport of important nutrients, oxygen, glucose. Blood flow is disrupted to the brain as well. So the blood-brain barrier and maintaining that integrity is very important. And both ApoE4 and type 2 diabetes lead to permeability of that. And so with type 2 diabetes, people have hypoglycemia, right? They have elevated blood glucose levels. And that over time leads to advanced glycation end products. These are basically like they cross-link proteins and, a variety of other things that, in the vasculature. And that basically damages the blood-brain barrier and leads to permeability. ApoE4, there's a variety of other mechanisms that happen, but essentially you can measure the permeability of the blood-brain barrier looking at a variety of biomarkers and proteins in cerebral spinal fluid, but also in plasma. And these have been shown by Dr. Slovovic, Dr. Montaigne to occur. It's like decades before the onset of, cognitive impairment. And it's literally, you can find it in more than 50% of all dementias. So it's happening independent of amyloid accumulation, tau tangles as well. So it's, either something that's happening well before. And in fact, blood-brain barrier permeability is also, the blood-brain barrier is essential for removing toxic compounds from the brain. And a variety of different processes, happen to allow this to occur. So, for example, you activate the glymphatic system during sleep, right? And a lot of people are aware of this. Your brain sort of swells during sleep, and the glymphatic system is pushing the cerebral spinal fluid through the brain, clearing out debris, amyloid plaques, things like that. You need the blood-brain barrier to be intact for that to occur. Yeah. So, that makes sense that basically if you have blood-brain barrier permeability happening, that you would start to have the accumulation of amyloid. So it's sort of like, and accumulation of amyloid.
I never thought of that, by the way, Rhonda. I'd never thought of what you just said, which is if your blood-brain barrier can't hold the back pressure, which is what would be the case if it were permeable, you would not have the back pressure to maintain the glymphatic flow. That never actually occurred to me till you said that. So very interesting mechanistic tie to that problem.
Right. And there's also like the other types of, and I'm not familiar with all the types of ways the brain, like there's other like parenchyma and stuff that are like, cleaning out the brain. All of those things are not happening as good when your blood-brain barrier, is dysfunctional, right?
And then another point is the ability of things to interrupt and get into the brain that shouldn't. So one of the things, everybody learns in pharmacology or medical school is that there are certain drugs that penetrate the blood-brain barrier. There are certain drugs that do not. The implication being certain molecules can pass through, certain molecules cannot. Well, presumably the leakier that barrier is, the more things that maybe we evolved to not have cross that barrier do indeed cross. And perhaps that's a part of what you just said, right? Which is this increase in inflammation that is now coupled with an inability or a decreased ability to clear out debris.
Exactly. In fact, so work from Dr. Montaigne has shown that fibrinogen, which is, you and I are familiar with this protein. It's something, if you are measuring if you're doing an inflammatory biomarker panel, right? So that's, it's a protein that's involved in blood coagulation, but it's also something that is a marker of inflammation, and fibrinogen is not supposed to be in the brain, but it's found in the brain in people with a leaky blood-brain barrier. And not only, so what's it doing in there? It disrupts a cell type called oligodendrocytes. These are a cell type that make myelin, that sort of fatty white structure that's important for, electric signals being fired throughout the brain. And it's basically toxic to them. So you basically start to have these, lesions and stuff in the white matter part of the brain, white matter hyperintensities, as you're probably very familiar with. Very common in small vessel disease. Also, you can see that in people with Alzheimer's as well. But getting that fibrinogen in the brain, like that's happening because it's allowed to get in there, and that so the permeability of the blood-brain barrier, again, like as you mentioned, basically preventing stuff from getting in your brain that you don't want in there. That's number one. But also the transport, I think you also alluded to this, you're not getting things like glucose into the brain. And in fact, all these transporters, they're in the endothelial cells. They're in these cells that are making up the blood-brain barrier. And when you start to disrupt that blood-brain barrier, those transporters are dysfunctional. So for example, one of them is the GLUT1 transporter. This transports glucose into the brain. As you start to get a disruption in blood-brain barrier function, glucose transporters, they go down. And so you're talking about not getting enough glucose into the brain, which is, again, that's one of the, pathological features of Alzheimer's disease, right? Not getting enough glucose into the brain.
That's actually an interesting explanation, because a very subtle point you made that might not be picked up on everybody is you mentioned the GLUT1 transporter as opposed to the GLUT4 transporter. And if my memory serves correctly, the brain has GLUT1s as opposed to GLUT4s, and GLUT1s are insulin independent. Is that correct?
Yeah, for the most part. I mean, they're insulin independent. I mean, that's to my understanding.
And why that's interesting is because it seems a bit counterintuitive that a condition that leads to insulin resistance, of course, type 2 diabetes is the essence of insulin resistance. It seems counterintuitive that would produce a hypometabolic state in an organ whose glucose transporters are insulin independent, except when you explain it the way you did, which is it's not the insulin resistance of the GLUT1 transporter that's the problem, the way it is for the GLUT4 transporter in the muscle. Instead, it's the actual mechanical disruption, if I'm understanding you correctly, of that transporter because of the way it's no longer presumably held in place by the barrier itself that allows glucose to get across. Is that, did I understand that correctly? Okay.
100% correctly. And I mean, this is, this isn't like dogma. This is definitely known. It's definitely where I'm heading. It's my opinion, but there is evidence of it. And I think it's time to explore this evidence a little closer and a little deeper, because type 2 diabetes, and you hear the term type 3 diabetes,
right?
And I think people think about it in the way of the brain being insulin resistant, and maybe there's something to that, but I don't know if that's exactly what's going on. I think the type 2 diabetes is disrupting the blood-brain barrier through a variety of mechanisms, including, the advanced glycation end products and the vascular. I mean, the vasculature disruption in type 2 diabetes is well known. I mean, they have all sorts of, problems, right? Retinopathy, the neuropathy, like all this. I mean, they are disrupting their vasculature, including these tiny, tiny little blood vessels that are smaller in size than are the diameter of a hair. Those things are disrupted at the blood-brain barrier. And when you disrupt them, their blood flows decrease and the transporters are going down. There's all sorts of problems. And so I think, fixing the diabetes obviously would be the, downstream thing to do, but it's kind of a new mechanism, right? Yeah. It's a kind of a new way of understanding it. And, so.
Right. In other words, it explains the observation. So there's an observation that is unmistakable, which is type 2 diabetes. I don't know the number, but I think it approximately doubles your risk of Alzheimer's disease. So in other words, even if you're sitting there walking around with 2 copies of the ApoE3 allele, having type 2 diabetes means you might as well have a copy of an E4 allele from a risk perspective. And what this is saying is, well, it's not entirely clear at the surface why type 2 diabetes would impact the brain through the lens of traditional thinking of GLUT4 transporters, which are insulin dependent. In other words, it isn't just an insulin resistance problem, but I think these 2 other things matter, right? What you said about the microvasculature. Again, what I think a lot of people don't realize is how destructive type 2 diabetes is to the kidneys, right? Because those tiny, tiny blood vessels, right? People are familiar with amputations and things that occur in digits because of that, impotence, right? All of these things where blood vessel is essential in small blood vessels. And so there's that which feeds into the vascular path that you've discussed, but it's also this disruption of glucose transport directly across that transporter. So I think it's actually a very compelling thesis for how type 2 diabetes could be acting via those two prongs to ultimately result in hypometabolism.
I do too. Yeah. And then of course the cascade of inflammation that happens after that. So, the other thing that's also very interesting is, and again, so we're talking about type 2 diabetes and very big implications there for the prevention of Alzheimer's disease, right? I mean, there's a lot you can do, as you've talked about, to prevent and even, treat with diet and lifestyle, right?
Yep.
Type 2 diabetes. But also what's really interesting, and, I was sort of on this trail years ago. I published a sort of integrative review on the role of the omega-3 DHA transporter, MFSD2A, in the brain. And what's really interesting is that animal studies, when you disrupt that transporter, it causes like 50%, breakdown of the blood-brain barrier, and like greater than 50% loss of omega-3 in the brain. So, in my opinion, it's that's animal evidence. Of course, there's human evidence where MFSD2A transporters decrease with age, particularly rapidly in Alzheimer's disease and with ApoE4. It leads to, so there are genetic, abnormalities and mutations that occur in that transporter where people have less of it, and they have microcephaly, so they have like smaller heads, and they also have cognitive dysfunction, sort of cognitive impairment, and things like that as well.
How do we know that, Rhonda? So that's let's even put the pathology aside, the sort of latter category, but let's just talk about. Well, again, what you've said explains something we've empirically felt is true, and the evidence suggests it. But this provides a mechanism, right? Which is ApoE4 carriers need a higher level of EPA and DHA to get the same benefit. That appears to be empirically correct. That would provide an explanation. Something else you said is almost like, we talk about amino acid or protein resistance, basically. Anabolic resistance as a person ages. They need more and more protein to get the same effect. It's almost like you're saying, if aging itself could create some resistance to dietary EPA and DHA that might require more as time goes on. Do you think there are also just genetic differences within the variant of normal, quote unquote, i.e. non-pathological, where one person would need more EPA and DHA to afford them the same benefit of protection as another person?
I do. I know there's at least a couple that are known. And so like some people have certain gene variants that actually they respond better to, for example, omega-3 supplementation, and others don't, where they would actually need a higher dose. And I think there's many more to be explored. Like we haven't unlocked all of that yet. When I say we, I mean the scientific community, not me personally. But I do, with the omega-3, and this again really hits home the preventative sort of role here that we can, have in our Alzheimer's disease risk. So with the MFSD2A, like these transporters are actually lost. So there's a type of cell called pericytes, per-E with an I, not to be confused with a parasite. They basically have these like big feet that wrap around the endothelial cells at the blood-brain barrier. And they serve really many important, but two main important functions. One is they're basically constricting and dilating and like helping squeeze like the flow of blood. So they're regulating blood flow to the brain, but they also are very important for that barrier. And they start to fall off with age, these pericytes, and inflammation plays a big role in that. But the MFSD2A transporters are concentrated on those cells too, right? And so you'll see like hotspots of where the pericytes basically once those pericytes start to fall off, that is when basically immune cells and everything starts going into the brain. It's like the start. It's like the start of the vicious inflammation cycle in the brain, of the leakage, amyloid accumulation, just everything downstream, right? And so there's something there with those transporters of omega-3 that are right at the same site, of where you lose those pericytes, which is also really interesting. And again, there's a lot of animal evidence that suggests the role of that transporter in blood-brain barrier integrity. Also, again, you can kind of like connect the dots here where you think, okay, well, this is a DHA, specifically it's DHA in phospholipid form transporter. So it's like, okay, there's got to be something here with the omega-3. And, I know there's a variety of scientists that are investigating this, but I'm sort of excited that I am now going to be part of a team. So I've joined the Fatty Acid Research Institute, which is Dr. Bill Harris's research institute, and as a research associate. And we've sort of secured a small grant to look at the role of omega-3 with blood-brain barrier integrity and biomarkers in people, a variety of different people that have small vessel disease, that perhaps go on to get Alzheimer's disease. And so I think there needs to be more research in this area, because the implications here, I think, are really important with omega-3 intake. I mean, it's one of so there's two main lifestyle interventions, I think, that are important with respect to the blood-brain barrier. Three. So not getting or fixing your type 2 diabetes. Mm-hmm. And then the omega-3 intake, and like defining that will be sort of tricky, but full stop, most people in the United States they're not eating enough fatty fish, and they're not supplementing with omega-3, which is sort of an alternative. And it was I think it was a 2012 study out of Harvard that identified low omega-3 intake from fish, so the marine type of omega-3, not the plant ALA, as one of the top 6 preventable causes of death. So it was up there with basically
smoking.
It was smoking and blood pressure and, obesity and being sedentary. Low omega-3 was it blew my mind. And they calculated some, I'm not a biostatistician, but there was some calculation done with estimating the number of deaths caused by not getting in enough omega-3 each year. It was the same number of deaths. It was 84,000 deaths a year from low omega-3 intake from fish.
I wonder, though, if that's also just a marker for poor health. I know that's the challenge of all of those studies, right? Is
totally.
In some cases with smoking, it's pretty obvious that there's causality there. I think there also is with blood pressure. But you could argue that never in the history of the world has there been a person who has, I'm being a bit facetious, who has, a high omega-3 index, who eats junk food and fast food all day, right? Those can't coexist. I want to ask one clarifying question, Rhonda. Certainly, I know that when you're talking about omega-3s, you're referring to the marine variant, of which we have EPA and DHA. But the transporter, if I understood correctly, is it a transporter only for DHA in its phospholipid form? And if so, what is the importance of EPA in this?
Great question. So the MFSD2A transporter that I've been referring to is specific to DHA. And DHA, the form of DHA is lysophosphatidyl DHA. And that, so that specific, we make it. And so we basically when we take in DHA from fish or from a supplement, our bodies, the higher amount of DHA that we take in, we add that lysophosphatidyl group to the DHA. We also have DHA in free fatty acid form bound to albumin. And albumin is not supposed to, that doesn't get into the brain, but it takes it to the blood-brain barrier, and the free fatty acid can sort of diffuse passively across the blood-brain barrier as well. Same with EPA. With respect to your question.
Oh, I see. But because the phospholipids on the DHA, it needs a dedicated transporter, whereas the the one that doesn't have a side chain,
phosphatidylipid
side chain can diffuse without a transporter.
Exactly. Yeah.
Okay.
So there's
All right.
that. So it's free fatty acid bound to albumin, and it can just diffuse across the
Okay.
brain. Yeah. So that's how EPA is generally getting in the brain.
And
But
then second question for you on that thread, and feel free to go into as much depth as possible, because I know this is actually a very important topic that is somewhat controversial. What do you see as the relative importance of DHA and EPA? The conventional thinking, I think, is that EPA probably more important in the heart, DHA probably more important in the brain. I'm sure that's a gross oversimplification, but can you expand on that?
yeah, I can try. I don't know that it's really known. So the way I personally think about both EPA and DHA, so there's a variety of metabolites and of DHA that are involved in resolving inflammation. So these are resolvins, the myrisins, the SPMs, protectins, and EPA also has some of those metabolites as well. And it also plays a direct role in inflammation through the sort of, I don't want to say inhibition, but like dampening the prostaglandins and the leukotrienes and a lot of the other inflammatory processes. So it's kind of like an approach where you're affecting inflammation from multiple ways, right? It's like a multi-pronged approach. And I mentioned fibrinogen earlier about like fibrinogen, it's an inflammatory protein. Well, it's involved in coagulation, but it's it's something that we do measure as a marker of inflammation. So there's studies showing that people that are exposed to particulate air matter, their fibrinogen goes up. But if they have a higher dose of omega-3 or higher intake of omega-3, it blunts that effect. Again, through the inflammation, right? So, both DHA and EPA are important in my mind for the brain as well. I mean, there's a variety of studies that have looked at even depression, and like you can induce depressive symptoms in a person by injecting them with what's called lipopolysaccharide, which is a component of the outer cell membrane of gram-negative bacteria. We have billions of those in our gut. In fact, there's about 1 gram of lipopolysaccharide, or LPS for short. It's also referred to as endotoxin. There's about 1 gram of that in our gut. Well, you can inject people with a low dose of that, something that actually would be somewhat, I would say equivalent to someone with intestinal permeability. And it can cause depressive symptoms in people compared to those given a placebo. And you can blunt that depressive symptom effect with EPA. Probably because of the the blunting of the inflammatory response. And there have been some, this is a field that's, again, understudied, underfunded, but, some preliminary evidence, randomized controlled trials, small randomized controlled trials that need to be, of course, repeated with larger sample sizes. They're basically showing that supplementation with EPA can help with depression.
Yeah, this is such a frustrating thing for me, and I obviously I know it is for you and for many others, including Bill Harris. If you took the cost of one phase 3 anti-amyloid failed drug trial, just take one of them, right? Like there's been dozens of them. Just take the dollars that were spent on one of those guaranteed to fail phase 3 trials, and put that money into a preventive trial that looks at something that's got real feasibility, or, something that's really interesting, like the optimal supplementation of DHA in the right patient population group, we could have an answer. And yet, for obvious reasons, there's an incentive to do a phase 3 drug trial on a candidate with an IND. There's not an incentive from a financial perspective to study these other things. And I think for a disease like Alzheimer's disease, that's particularly problematic because as I suspect we'll discuss, unlike cardiovascular disease, where yes, prevention is still the best strategy, you can come in late to the game and still make a difference. I don't think the evidence is particularly compelling that that is true for Alzheimer's disease. Now, I'd love to be wrong, but I have yet to see compelling evidence that you can be a Johnny-come-lately to that pathology and have an impact.
it's hard to fix those leaks in the brain once they're started, right? I mean, and that also is why I think there have been failed trials also there've been a few with omega-3 supplementation in people that already have Alzheimer's disease. And you're giving them like, I don't know, at most 2 grams.
Right.
I've seen studies like 500 milligrams. I'm like, are you kidding me? Like you know, patients with high triglycerides or cardiovascular problems
And
are
we're giving them
prescribed
4
at least
grams.
4.
Yeah.
I know, at least 4. You know, this is something that has the safety of a nutrient, but literally can act like a pharmacological drug, you know, at higher doses. And so I agree with you. I think it is much more challenging to fix, you know, when you have Alzheimer's disease. And certainly, like, I'm talking about like the leaks in the brain, but like, then what happens after that? The amyloid accumulation. And like, when you start to get to this level, when you've got all of that, I mean, good luck. It is going to be challenging, and you're going to have to take, it's not going to just be fixing the amyloid. You're going to have a cocktail that are going multiple angles, I think, in order to get some improvement. I think the amyloid and perhaps also fixing the blood-brain barrier leaks as well, maybe at the same time with the cocktail may help a little bit. But prevention is the way to go. I mean, like, it's so much better to not get Alzheimer's disease than to try to fix it once you have it, because it is a very complicated disease with lots of things going on. So, you know, I think that the strategies that can be done, and they're not that difficult, right? So we talked about type 2 diabetes. I mean, there's no reason why someone should have it. Like, right? I mean, like, you should be able to.
Yeah, I would phrase it as, I do not believe for a moment that type 2 diabetes is inevitable to our species. Whereas I do believe atherosclerosis is inevitable. Of course, I also think most people don't need to die of it. So that's a very stark contradiction. The disease is inevitable. In as much as humans will have lipoproteins that carry ApoB, we will get atherosclerosis. But again, we have the technology to delay the onset of that disease to the point where it should not be the cause of death. We should be dying with it, but not from it. I would also argue that cancer is inevitable to our species. It is simply a stochastic problem where if you live long enough and if you accumulate enough genetic mutations, and we can do lots of things to reduce the risk of that and to delay the onset of that and to detect cancer early and be more successful in treating it. But the incidence of cancer strikes me as something that is inevitable with enough age. I actually don't feel that way about type 2 diabetes. In other words, I think I share your point of view, which is it's not something that is necessary. We don't have to eventually get it. And I think that makes it all that much more tragic that you watch how many people are suffering from this disease and how much damage it's causing, not just in the disease itself, but as you said, it's such an amplifier of what I refer to as the horseman, right? It's what it does to your risk of cardiovascular disease, cancer, and Alzheimer's disease is actually why the death toll for type 2 diabetes is so grossly underappreciated.
Yeah, it's accelerating the aging process, right? I mean, like the molecular.
Yeah, it's gasoline on the fire of aging. I'd have to really reflect on it, but I can't think of a process that accelerates aging more than type 2 diabetes.
Right. I mean, and maybe morbid obesity, but like they're probably also type 2 people.
I would argue only in the context of insulin resistance, which gets us right back on that path. Yeah. So,
right.
So I mean, I think the good news there is not that it's easy, but it's like we sort of know what it takes to treat it and prevent it. And it doesn't necessarily look like the strategies that are being deployed, unfortunately, at the level of the ADA. I want to ask you one other thing that we haven't talked about, but I want to know if it pertains to the blood-brain barrier at all, and that's blood pressure. So hypertension and I guess hyperlipidemia also pose enormous risk for not just cardiovascular disease, where they are 2 of the 3 biggest drivers of risk, but they also pose a risk in Alzheimer's disease. And I wonder, do either of those act specifically through the blood-brain barrier?
It is another really important modifiable lifestyle factor that can affect Alzheimer's disease risk. And, so blood pressure, maintaining good blood pressure. So, I mean, basically you want to be systolic below 130, right? I mean, like once you get to 130,
the sprint trial would even say 120.
120, yes. Yeah. And the blood pressure itself, so getting that blood flow to the brain, blood-brain barrier specifically, is so important. When you don't have, that when that blood flow doesn't, basically isn't able to get to the blood-brain barrier well enough, those tiny little vessels start to like just fall off. And it's one of the reasons why exercise also is so, so important. So yeah, blood pressure. So there's been a variety of studies that have looked, of course, as you mentioned, the observational data is never able to establish causation, but it's still an interesting point to look at in combination with many other types of data, right? But
especially when it's always in the same direction. I mean, I think that's what that's what differentiates the epidemiology around, for example, exercise and blood pressure from the epidemiology around nutrition. The epidemiology around nutrition, it has very low hazard ratios and it's always changing the direction it's moving in, suggesting that whatever's being studied probably doesn't matter. Yet when you look at, the epidemiology of smoking, blood pressure, dyslipidemia, exercise, much bigger hazard ratios, virtually always pointing in the same direction. So it strikes me that the latter is signal, the former is noise.
Right. Good point. Yeah. So blood pressure is associated, particularly, 50% of people, adults in the US have hypertension, and about 20% of young adults, like we're talking people age 18 to 39, have hypertension. Like that's crazy, right? And it's actually the high blood pressure. It's the cumulative exposure to high blood pressure that's really damaging the vasculature. And so it's the younger people, it's the people that have it earlier in life that should be the most concerned and are the least, right? Those are the ones that are like, I'm young, I can worry about it later. But yeah, so high blood pressure is associated with dementia risk, particularly when you have it like before, the 50s. Like, so when you get it in your, before the 50s or, midlife, once you start to get high blood pressure in older age, like 70, 80, it's not as much associated with, the Alzheimer's disease and dementia risk. So it really does seem like cumulative exposure is the key factor there. But, again, one of those things that, is a lifestyle factor that's easily modifiable. Exercise improves blood pressure. Sauna improves blood pressure, right? Those are two, basically low-hanging fruit lifestyle interventions. Some people do have gene polymorphisms where they're very sensitive to salt intake and sodium intake. And that combination of those people with like a higher sodium intake really seems to skyrocket blood pressure. It also, I think, is one of, like you were mentioning nutrition, I think it's also looking at the combination of genes and diet. Most nutrition studies don't do that. Like they don't. And there is an interaction going on. And I do think that's why some of the sodium intake blood pressure literature is just a little more, I would say.
Yeah, it's all over the place.
Complicated.
Yeah.
Right.
I wanted to ask you a question about exercise, but before I do that, I want to actually pick up on something you just said. How optimistic are you? I don't know, let's bracket this with, let's say in the next decade, that we will have more of a sense around what precision nutrition looks like as it pertains to genes and polymorphisms of them. In other words, people talk the talk like, oh, I did this test and it told me I should be eating this, that, and the other thing. But the reality of it is there's nothing that's available today that's come close to offering that type of insight. Do you believe that that type of insight is available? You used one example, which I would agree with you on, which is that there are probably different levels of genetic susceptibility to sodium that might suggest one person needs to be eating 2 grams a day of sodium and another person needs 4 grams a day. But do you think it will get further than that? And how likely do you think we will be to extract that information?
I do think in 10 years, we're going to know a lot more about, precision nutrition, obviously precision medicine as well, because there's also an interaction between, pharmacological treatments and genes as well, as you know. So, in 10 years, we're definitely going to be a lot further than we are now. The problem is, and this is always the problem, we've already alluded to it a few times, is the incentive for funding to study those things that aren't necessarily going to be super profitable. It's, the government, the NIH, there's a certain amount of funding that you can get from them. And they often like to study one sort of thing. It's hard for like the nutrition when there's like multiple things involved. It's like, they're just like too complicated, And so, there's a lot of funding from pharma industry, for example, and then they put in lots of money because they are incentivized to do that when it's a drug, right? So that's my one concern with I would say, gene diet interaction when it's more on nutrition. But there are people that are it's a growing field of research for sure, and I do think there's technology is advancing too. I mean, so at the point that our technology is advancing with AI and stuff, I think we're going to start to see an exponential there, honestly. So I am optimistic that in 10 years, that it's going to be a lot easier to sort of delineate what a person should eat based off of, the genetic makeup, versus just what, generally we think is healthy.
And do you think that will be at the level of macronutrients or micronutrients? Like what do you think, how much heterogeneity do you think there is among people as it pertains to factors like that?
I think both, for sure. I mean, there are differences in the response to macronutrient intake and the response to micronutrient intake. And there are people, like there's vitamin D polymorphisms, right? Where people, for whatever reason, they actually so maybe they evolved in a place where there was so much sun or something. I don't know what the cause is, but like some people, they have to supplement with high doses of omega-3 to get to be able to convert the
vitamin
precursor
D.
to the hormone, right? The steroid hormone. That's just one example. There's selenium. I mean, there's magnesium. There's a lot of different micronutrients that are also, and there's omega-3. There's omega-3 as well.
And B vitamins.
Exactly. There's B vitamins. Yeah. So I do think that it will kind of, more people are looking into it. And again, with advancement in technology, things will become cheaper and easier to do. And that's going to create an exponential, in my opinion, where it's like, okay, maybe in a couple of years, we'll start to really see an explosion. And then after that explosion, exponential happens where people are building off of that because, that's how it works, right? So that's kind of where I think it's going. And I'm excited about it. I think that's where it needs to head. I think it's going to clear up, as you mentioned, all the conflicting data with nutrition. I mean, nutrition studies are a mess. Designing the right trial. I mean, the part of the reason for that is because we have evolved these genes. I mean, when it's a drug and you don't have, I mean, with the exception of, yes, there's the CYP enzymes and stuff that help us metabolize xenobiotics, things that are not a vitamin or a mineral or essential amino acid or fatty acid, right? Something that's foreign to our body. But by and large, when you give someone a drug, like they're starting with 0 levels of that drug in their body and you give them the dose that you're giving them, it's like clear you're giving them a dose and it's going to be different than people getting a placebo, right? It's going from 0 to something. Whereas when you do this nutrition study, a micronutrient, you give them a vitamin or a mineral, nobody's starting with 0 for one. Your placebo group could have 5 levels of that. And unless you measure something, you'll never know, right? So it's very different and all the trials are sort of, they're trying to mimic that gold standard of a randomized controlled trial with a pharmacological drug. And it's just, you have to like put in so much more effort with nutrition. With the drug, you don't have to start doing blood samples of this and that and measure the drug and make sure people aren't deficient in that drug. But of course they're deficient in the drug before they start the trial. It's like a drug, so yeah.
It's such a good point, Rhonda. And I'm going to use 2 examples to highlight why this is so important. And because it ties to 2 things we've been talking about, right? So if you look at how a blood pressure trial is done, it is exactly what you say. It's titrated, meaning it's done the way a pharma trial is normally not done. So a blood pressure trial says, let's just bring in a whole bunch of people with high blood pressure. Okay. You guys on the placebo or on the low treatment arm, we're going to manage you to a blood pressure of 140/90, but no lower. You guys in the treatment arm, we're going to manage you to 120/80 or better. And by the way, we're managing to the outcome, not the drug. So I'm agnostic as to whether this person needs a higher dose or a lesser dose. We're checking the outcome here. And so if we did a vitamin D trial that way, it would be a very different trial. And one of my biggest criticisms of vitamin D trials, and why I think we don't have an answer as to whether supplemental vitamin D is valuable, is they don't do this. You take the people, you divide them into two groups, you give one group a placebo, you give one group usually a very low dose, somewhere between 2,000 to 4,000 IU daily. But we don't actually know what the level goes to. In other words, a better vitamin D trial would be, we take a bunch of people whose vitamin D is below 30. In one group, we give a placebo, and in another group, we give whatever it takes to get them to, I'm making this up, but 80. Then we would see, is there a difference between this group that's below 30 and this group that gets to 80, regardless of the dose it took to get them there? Because of course, that may include part of the problem. So I think your point is an excellent one. And I think it's something that listeners need to be aware of when they're scrutinizing trials of this nature, which is a negative trial doesn't mean the thing doesn't work if the trial wasn't designed correctly.
Right. And it's a matter of money too, right? Like to measure all those things, it's cheaper to just give someone the vitamin D supplement and then look at the outcome and go, oh, didn't work. And then you have another confusing piece of literature out there that people are like, oh, but it's, it says that vitamin D supplements do nothing. Yeah, it's a big problem. And I think as, our technologies are advancing, that's going to become less of a problem as well. So, at least I hope. I guess you never know, but yeah.
So I want to go back to something you talked about earlier, which is you just touched on exercise. And I want to kind of visit now the suite of things that exercise does, because people who listen to this podcast know if there's one thing I just can't stop talking about, it's exercise. But I think there's a reason for that, right? It's not just that I love exercise. It's that the evidence is overwhelming that a person who exercises, especially at the right amount, right? Like we're talking not just 30 minutes a week type thing, but if you're really doing the work, you're having a greater impact on the reduction of risk of Alzheimer's disease than any other intervention you can take. Now, there are lots of interventions that matter. Sleep matters, nutrition, as we talked about, type 2 diabetes, all these things matter, but the risk reduction that comes from exercise is enormous. What do you think are the mechanisms by which that is happening? Because I suspect there's many.
Oh, definitely. There's definitely many mechanisms happening. And I would, first of all, I 100% agree with you. Like there's nothing better than exercise. And of course, any exercise is better than none. But for me, because as I mentioned at the start of this podcast, I'm very focused on neurodegenerative disease. I specifically sort of design my workout routine based off of what I think are going to give me the biggest brain benefits. And what I've sort of come to the conclusion of is that intensity does make a difference with respect to the neurobiological effects, exercise intensity. And there are some mechanistic reasons for that. But just talking about like what's moderate intensity exercise, what's considered vigorous intensity exercise, right? If you look at some of the recommendations out there by the committees that have, there's a team of scientists, physicians that sort of analyze all this data and then make these health recommendations based off of that data. It's 150 to 300 minutes of moderate intensity exercise, right, which I think they define as like 50 to 70% of maximum heart rate. In vigorous intensity, they say 75 minutes, I guess, a week, and that would be more of you're getting above the like 75 to 85% max heart rate. So that's what they I think define as something like that, vigorous. And they have a variety of examples. There's the World Health Organization, and there's a variety of committees that are they come to that same conclusion. For me, I like to go higher intensity. And this is something I kind of was looking forward to talking to you about, because it has to do with how do you measure, so there's the estimated heart rate, which is what I do. I've estimated. And again, all sorts of problems with that based on your physical
Meaning
fitness. Right?
estimating your maximum heart rate to take a percentage
Yes.
of that? Okay.
Right. Yeah. Right. I mean, so basically, like if you, the more fit you are, like you could be doing a more vigorous intensity exercise, but your heart rate like doesn't go as high as someone who's not fit. Right. And then people that are older. So there's, of course, I would say problems with that, but generally speaking, that's one way. But I'm also very interested in lactate, and I know you are as well. For me, I actually want to get my lactate levels high, like high. Yep. And the reason for that is the neurobiological benefits. So lactate, as you know, was once thought to be this sort of end metabolic byproduct with no, like, it was like, useless. Well, not only just useless, actually it was thought to be harmful, right? So
at least to performance. Yeah.
Right. Harmful to performance with respect to people thought it was like causing their muscle soreness, right? And that's sort of, as you know now, that it's not lactate, it's the proton buildup, right? So lactate is sort of in homeostasis with lactic acid, and it's the protons that were sort of responsible for that.
And neither of them are responsible for the soreness you feel the next day. That's the micro trauma, of course. Like the soreness and the burn from the hydrogen ion is gone, minutes after you stop exercising.
Right.
Yeah.
Yeah, exactly. So the lactate that gets into, so you're generating lactate when you basically are pushing your mitochondria inside your muscle cells to a point beyond where they can generate enough energy in the form of ATP, adenosine triphosphate. Then they sort of have to figure out another way, like your cell has to figure out another way to get the energy, right? So this is where glycolysis comes into play. So this is happening outside of the mitochondria. And I'm sure your listeners have heard all this before, but for those that haven't, the lactate generation is then from that, metabolism of glucose outside of the mitochondria. And that's happening when, again, you reach that threshold of, basically you're pushing your muscle cells hard enough and their mitochondria can't keep up with, producing enough energy. So the lactate itself, and these studies date back to like the 70s. It's been shown that lactate, that it gets into circulation and it's used by other organs as an energy
fuel.
source for one, as a fuel. Yeah. And the brain being a big one. And this is now, this has been, decades of research, but Dr. George Brooks has, he was like one of the first to propose this lactate shuttle theory. And he's, of course, provided evidence for that, as many others have as well, where, during exercise and after exercise, the lactate that is generated, from muscles that gets into circulation is consumed by the brain. This has been shown in humans and animal studies, of course, but it's consumed by the brain. And also, not only is it consumed, it's acting as a signaling molecule and increasing at the blood-brain barrier. Lactate itself has been shown to be responsible for the production of what's called VEGF. This is it's a vascular endothelial growth factor, VEGF. And what it's doing at the blood-brain barrier is it is growing new vessels and repairing damaged ones. So that brings it back to what we were just talking about, right? The vascular damage at the blood-brain barrier. Lactate itself is a signal to increase that VEGF. It also increases brain-derived neurotrophic factor, BDNF, at the brain, at the blood-brain barrier, and in the brain as well. And so,
where is BDNF produced?
Many places. It's produced in the capillaries and in the vascular system, in the heart, in muscle. It's produced in the brain. And so exercise increases brain-derived neurotrophic factor in many different parts of the body, in many different organs and tissues. And it's really interesting because there's some evidence that the sheer force of blood flow, so essentially like the more vigorous you are exercising, your heart's pumping, right? And your blood is just going faster, right? It's moving faster. Well, that sheer force on the actual endothelial cells lining the blood vessels is a signal to increase BDNF as well. Super interesting stuff there. But it also increases BDNF, in muscle, which it plays a role in repairing damaged muscle. It increases in plasma. brain-derived neurotrophic factor, although there's a little controversy about this, it can cross the blood-brain barrier, and it also is produced in the brain as well. So all of those things, and brain-derived neurotrophic factor is really important for many reasons. I mean, it's important for long-term potentiation. It's important for neuroplasticity. So long-term potentiation is, basically strengthening the connections of the synapses that are connecting neurons. It's involved in basically long-term memory retention, but also it's important for neuroplasticity. So brain-derived neurotrophic factor plays a really important role in that. And that is also something that decreases with age. And it's essentially, I think a really easy way to think about it is your brain's ability to reshape and restructure with the changing environment. And like as you're aging, things are changing in the brain. And you have to be able to respond to that. Your brain responds in a plastic way. And that happens very well when we're younger, not so well when we're older, not so well in certain disease states like depression, neurodegenerative disease. And so that brain-derived neurotrophic factor plays an important role in that. But I am going for higher lactate. And so I, much to your, basically based off of your recommendation, I got the lactate meter, the Nova, the one that you have. And I've been using that to kind of try to get my lactate higher than typically what I think you are doing with your a completely different type of training.
Well, we do kind of both ends, right? So on the one end of the spectrum, what we want to do is increase our mitochondrial capacity to maximize aerobic metabolism. And there are two ways to do that, meaning you have to do two things. So the way I describe this to people is the way one of my coaches described it to me when I was a fledgling cyclist. Your aerobic capacity is a pyramid, and the area of that pyramid is your total aerobic capacity. And to have the largest area of a pyramid, you need the widest base and the highest peak, right? A pyramid with a narrow base and a high peak, not as good. A pyramid with a very wide base and a shallow peak, also not great. You want wide base, high peak. Well, in that analogy, the base is your zone 2 threshold, right? It's how much work can you do while keeping lactate at that sort of threshold that George Brooks and Inigo San Milan talk about of about 2 millimole. So what differentiates the best aerobic athletes from someone, say with type 2 diabetes, which would be the opposite end of that spectrum where you have real metabolic dysfunction or rather mitochondrial dysfunction, we're talking a 4-fold difference in watts per kilo output. And you just have to train at that level. You have to get to that threshold and train right there. So, this morning, that was the workout I did, right? It was a zone 2 ride where I'm just riding right at a lactate level of 1.9 millimole was where I was today. But you do need to do what you're describing as well. You have to do the pyramid building. You have to build the peak of that pyramid. Those are the VO2 max sets. And generally the sweet spot for building those is 3 to 8 minutes of all-out effort for the respective duration. So obviously what you can do for 3 minutes and no more is harder than what you might be able to do for 8 minutes and no more. My favorite are 4 minutes. So for example, on Sunday, that was my workout. I actually did an hour of that zone 2, kind of 2 millimole stuff, and then did 4-minute awful repeats where it was much, much higher power for 4 minutes. And then I rested for 4 minutes and then went again for 4 minutes and then rested for 4 minutes. And at the end of those 4-minute blocks, my lactate will be 15 or 16. And,
wow.
And then at the end of a 4-minute rest, it might be down to 6 or 7. And then we do it again and do it again and do it again. So yeah, long-winded way of saying you want both, right? You want to build that pyramid to be as wide and as tall as possible. What I don't think I appreciated though, was that the brain is getting a benefit from those lactate peaks.
Yes, it's getting a benefit, and they don't it's cleared quite quickly.
Yeah.
So, it's
minutes.
It's minutes.
If you do nothing, if you just stopped, and of course, athletes are even better at this, right? Like an amazing athlete would clear lactate within, they'd go from 10 millimole to 2 millimole in minutes.
Yeah. For me, yeah, like, 20 minutes later, I'm back to my 0.9, millimole baseline. But, that lactate also it's important for neurotransmitter synthesis. You're making glutamate, the major excitatory neurotransmitter in the brain. It's important for making precursors to that. Norepinephrine. I mean, these have all been shown in human studies, also animal studies. So for me, I do a lot of Tabata training. And
so that's even more intense because you're only doing 20 seconds on and 10 seconds off. So that's really intense. Yeah.
It is. I do, and I do 16 of those. So I do, like 8 and then separated by like a 30-second, break. And then I do another 8. And
so two 4-minute blocks.
Yeah. And then, well, it's a total of about 10 minutes. But so the first minute I'm in like zone 2, and then by like the end of that minute I'm like zone 3, and then I go into zone 4. Like, so it's like.
And what are you doing this on? Are you doing this on an air bike?
Peloton. I'm doing it on a Peloton, okay. Right? And my Apple Watch is beaming my heart rate and my zones onto my screen, which is kind of, they're all estimated, of course. Again,
yeah.
So, but I do that five days a week. It's, again, I think. For me, the efficiency also, so I'm trying to maximize the neurobiological effects for me with exercise. And I really find pretty compelling evidence that intensity is really important with respect to that for the brain. Not that there isn't a benefit for, lower intensity exercise, but it's certainly people doing, moderate intensity, like the more time you put in, like the volume of training, right, is like, you're going to probably find some equivalent there, right? But with the lactate, though, that's the one mechanism I'm like, that is really something I personally am trying to optimize for. And it's definitely a consequence of intensity, right? But the fact that it's cleared so quickly, and it's transient, like I'm wanting a lot of it, like each day. I'm wanting to keep doing it, right? Like, so I kind of, I do those types of training.
But there are other ways to do it that you might want to consider, right? So you might want to say, look, I'll do those Tabatas 2 days a week. I'll do some longer, slower cardio. And to be clear, like when I'm doing that zone 2, it's not trivial. Like, I mean, my heart rate's still 140 when I'm doing that zone 2. But what you could do is use blood flow restriction when you're lifting weights on the other days, and that will get your lactate through the roof. So 4 days a week, I'll do blood flow restriction at the end of every workout. So 2 days will be upper body, 2 days will be lower body. And then especially on the lower body days, so you've got these huge cuffs at the upper part of your thighs, and I'll do, leg presses, leg extension, leg curl, and finish up on an air bike. And by the time you take those cuffs off, all that lactate that's been pooling in your legs for 10 minutes will flush through systemically, and your systemic lactate level surges. So
what do your levels get to? What do your lactate levels
I mean,
after?
Not as high as I would get on an all-out sprint. I mean, like the highest I've ever had my lactate is about an 18 or 19, but I can still hit the mid-teens doing blood flow restriction. So
that's incredibly high.
Yeah. It's funny, when I used to coach athletes, I worked with an Olympic swimmer who could get to 26 and still be conscious. I mean, I say that sort of half jokingly, not that too much lactate would render you unconscious, but the pain that you must be in when your lactate is 26 is comical to me. But he could finish a swim race like something like a 400 individual medley, which is, probably the highest lactate generating race there is, because it's all out upper body, lower body assault, have a lactate of 24 to 26 millimole, and 4 minutes later jump in the pool again and do a race and enter the pool with a lactate of maybe 6. It's pretty amazing.
That's amazing. What, so
my point is, like, you could diversify the training a little bit. And if you're still, because again, you'd still get that lactate hit, but you'd also be diversifying the training. Because I do think it's hard to make the performance gains if you're really doing an all-out Tabata 5 days a week. I think that is hard.
What kind of performance gains are you talking about?
Within the Tabata itself. In other words, it's hard to make gains on the power output that you want to be generating. Because that's when you're doing 20 on, 10 off, right? You're trying to get as much power as you can in those 20 seconds. And those are what we call match burning workouts. Like you're burning all the matches that day, right? Those are.
Yeah. I'll tell you something that's interesting. Have you ever tried doing an aerobic or a high intensity workout with like a mouth tape where you're just breathing through your nose?
Mm-hmm.
You're
limited, of course. I mean, at some point you're not going to get to you're going to be limited by, depending on, for me at least, I can't do my all out best without mouth breathing at the end. Once I reach 215 to 220 watts, I can't sustain maybe 225 watts. I need to start breathing through my mouth. I need to at least every other breath use my mouth. And I would expect everybody's sort of different there.
Right, exactly. People are different. I mean, different nasal, like different sinuses and shapes of your nose and everything. I just recently started doing it, and it was kind of odd because maybe I'm not going, like I PR'd the first time I did it. And I didn't, I wasn't going as hard on my all outs, but I think on my rests, I was going harder on my 10 seconds off. I was like not really bringing my resistance down.
Have you tried doing it where you do nothing on the off? A pure off?
What do you mean? Just like keep going?
Don't spin whatsoever. So do the 20 seconds all out, and in the 10 seconds, you're not spinning at all.
Oh, I've never tried that.
Give that a try. Well, I think the goal is to make the hard as possible. And so being truly off for 10 seconds will make it more likely that you can deliver the maximum wattage during the 20 seconds.
Okay. I've been trying to do the opposite, where I'm like, okay, on my off, I'm like, zone 3. I'm really like, I'm still putting in quite a bit of power, right? Like
that's good too. I don't want to say just that there's, those are, we used to call that sweet spot workouts, where you would go zone 3, zone 5, zone 3, zone 5, zone 3, zone 5. But what you really probably want to be doing in a Tabata is zone 6, zone 1, 6, 1, 6, 1.
What's zone 6?
It depends
just
on
like
the system.
Maximal?
So, basically, so technically the literature on this would suggest that we are only able to hold full maximal effort for 10 seconds. Anything we do that's longer than 10 seconds, we are applying some governor to the system. Now, so at 20 seconds, you're even if you don't realize it, you're somewhat pacing yourself, even if you're trying to go.
Yeah, I'm not doing maximal. I'm not for sure. Like my husband, like he seems like he really gets to that all out, but I'm not for sure. I'm definitely not maximal at my 20 seconds. No way.
Yeah, that makes sense, because I don't think one could do that 5 days a week. I think you would. I think you would fry yourself.
What's also really interesting, Peter, is that though, like by like the 4th or 5th day, I'll still I'll be PRing, so I'm always competing against myself. And
by what metric? By wattage?
Oh, the Peloton. No. I'm not nearly as scientific as you.
Okay, so what metric?
So Peloton ranks you based off of.
But isn't it based on kilojoules or watts? Isn't it based on average wattage?
It is.
Yeah. Okay.
Yeah, but I don't know all those off the top of my head.
I see. Got it.
You're
Okay.
writing everything down. I'm not doing that.
Yep.
Yeah. But my lactate.
But that doesn't surprise me, Rhonda, because the way to get maximum wattage is to hold your highest constant maximal wattage and go. So zone 4 held indefinitely will produce a much higher average wattage than zone 3 alternating with 5 or 6 alternating with 1. So yes, if your metric of success is what is my total average wattage over the course of this workout, it will not be a Tabata. It will be a steady state. All I can, that's actually what's called FTP, functional threshold power, which is what technically the Peloton is using to estimate your zones. Have you ever done the FTP test on the Peloton?
No. Mm-mm.
So that's actually probably worth doing. So if you go into the Peloton, there's something called, I think it's called fitness test. And it's going to have you do either, you get to pick 2 8-minute all outs separated by some rest, I forget how much, or 1 20-minute all out. I prefer the 20-minute all out. I think it's a better test. It will take your average wattage over 20 minutes. It will multiply it by 0.9, and it will say that is your functional threshold power, which is defined as the maximum power you can hold for 1 hour. And in cycling, and Peloton uses this system, that is the metric by which the zones are set. So zones 1 through 7 are a function of power, not heart rate. And they are all a function of that FTP number. Now, again, none of that's necessary for Tabata, right? Tabata didn't rely on knowing those zones. It wasn't about titrating to a given heart rate. It was simply a question of go as hard as you humanly can for 10 seconds and then do nothing for 10 and do that 8 times. So
yeah, I'm definitely using a different.
You're using kind of a slightly different protocol where I think like you're doing a workout where you're going much harder during the rest and then not as hard during the workout because you wouldn't be able to.
Yeah, it's definitely right. No, I'm definitely not doing it the way I guess.
And how high are your lactates? And you check your lactate at the end of the both sessions after the 2 rounds.
After so the 2 round, it's all like a continuous thing. Like it's you get like a 30-second rest period between the 2 8 sessions, but like I'm not resting.
Yeah, you're going hard.
I'm like probably zone 3. Yeah. So at the end of that, now my lactate doesn't get nearly as high as yours, but I'm typically like around 7 to 8 millimolar. So that could give you an idea of my I'm what I call a committed exerciser. I'm not an athlete as I would consider you are, I'm sure.
Well, actually it's actually quite different. There's the best athletes in the world, like world class, right? So both Michael Phelps and Lance Armstrong, you could argue 2 of the greatest, both actually put out relatively low lactate levels. So you don't know, you might be one of those people who's so efficient that you don't actually make much lactate. So it's not, it's actually, like I don't think Michael Phelps has probably, even when smashing world records, probably ever seen a lactate above 10.
Yeah, but I'm definitely not one of those guys. I'm not an athlete. Well, it would be interesting to differentiate for sure. I mean, but like I do know, for example, so when you are doing at least a high intensity interval type of training, which I would say that this, would you agree this type of training definitely would fall into that? And of course, people generalize this term and stuff, but that's a whole other issue. But like that does, like you are sort of forcing adaptations on your mitochondria to make more mitochondria, right? Because your body's like, oh, I no longer can use my mitochondria to make energy. I got to rely on this other process, glycolysis. So as an adaptation to that, you increase mitochondrial biogenesis. And that's been shown now in several studies, in human studies, right? That you can increase mitochondrial biogenesis. Now, also aerobic training does that as well, but you can
it's really a question of time. I mean, to me, this is what it sort of comes down to what you were saying earlier. So if somebody says to me, I've only got 10 minutes a day to devote to aerobic training, what does it need to be? Well, the answer is clearly it needs to be the type of training that we're talking about here. Now, if someone says to me, I don't want the minimum effective dose, I want the maximum result, then I'm going to say, well, would you be willing to give me 90 minutes a day? In which case we could build you the biggest pyramid, basically, right? I need like an hour. Yeah. I need an hour a day to build you a mega pyramid. But some people don't have the time or desire or interest to do that. In which case, we would need to just get people to doing, I get asked this question all the time. I think in 10 minutes a day of cardio and, probably 30 minutes a day, 4 times a week with strength, you can get amazing results, but you have to be laser focused and there's no messing around. Like it's, I'm sure when you're done that 10-minute workout, there's no ambiguity about how hard you've worked.
No, not at all.
So it's not, it has to be very vigorous. Yeah.
It does. And I do also use Peloton for my strength training as well. And there's a lot of like paired sets and supersets. And so it's like nonstop, training where it's definitely like I'm putting effort in, but it's not a long session either. And I do try to do that. Like it, I'm probably putting in the minimum amount of strength training that I can personally do, which is like 40 to 50 minutes a week. And I used to not do any, so like even that's like,
all right, we have some
progress.
stuff we're going to talk about at dinner here because I'm gonna try to talk you into doing a little bit more, but I want to ask you, where do you put your sauna in relation to this? Do you dissociate in timing sauna from your exercise? Do you go right into the sauna after you work out? How do you incorporate that?
It varies. So I do both a regular dry sauna. Actually, I don't use it as a dry sauna. I do a lot of steam as well. But I do that, but I also do hot tub. So I do a jacuzzi as well. And both of those, forms of heat stress have been shown to increase, heat shock proteins, which is sort of a biomarker of heat stress. And both of them also have been shown to increase brain-derived neurotrophic factor as well. So heat, I think, also plays a role in that like stress response. But it depends on the day. So I often will, in the sauna, I like to read scientific papers or listen to podcasts like The Drive, or like I'll listen to like if someone's on Tim Ferriss's show. Like there's only a couple of podcasts that I ever listen to. Yours is one of them. So it's not like that's like my time where I'm like, because there's no other time. Like if I'm in the car with like my child, like most of the time I'm listening to, Frozen music or whatever, like it's not, I'm not listening to The Drive. So, so it's like it depends on what I'm doing, but also I like to do hot tubs at night. So typically the sauna will be in the day. So I do my workout in the morning. I'll have the sauna warmed up and ready to go, and I'll get right into the sauna after my workout. And I either have a paper in hand or I'm going over a presentation or something. I find it's really interesting. I don't know if you've ever tried this or observed it, but this goes way back to my, days as a graduate student when I first started using the sauna. I realized that like if I would go over a talk that I was going to give, like a departmental meeting or whatever, I was giving a talk. If I went over it and thought about like what I was going to say in the sauna, man, did I remember it better. Like it was like very clear that there was something going on with my memory. And I mean, very, very consistent. Of course, you know me, I like was diving into the literature. I'm like, there's got to be something to explain this. And, lo and behold, there's like a variety, like there's like certain growth factors that you make that in the sauna with heat stress that do affect like memory. So, plausible, hypothesis there. But anyways, so if I have something going on, like a podcast or a presentation.
What's your protocol? What's your temperature and duration in the sauna?
So typically, I like to, it depends on how hard I went on my workout too, or if I'm doing it like right after the workout, or if I'm like midday, just like, I'm going to take a break from what I'm doing at my computer and I'm going to go read a science paper in the sauna. So like, it really all depends. Generally speaking, if I go in right after I do my Tabata session, I probably stay in about 20 to 25 minutes, and my temperature is like 175 degree Fahrenheit. Okay. If I am not going in right after a training session, then I'll stay in longer. I'll stay in probably a little bit longer than 30 minutes. Sometimes I'm pretty adapted to, and my temperature will be 175, 180 sometimes. I also do the humidity, which makes it feel hotter as well. So I guess anywhere between 20 to 30 minutes, and my temperature is anywhere between 175 to 180. I find that I used to do really, really hot, and I just sort of like about like 190, like I was doing 190, and it was giving me like my head, I was getting headaches more easier. And so I didn't, I just didn't like it, and I didn't feel good. I was getting dehydrated.
How long did you sauna during your pregnancy? How far were you able to and what do you I'm sure women ask you this all the time, and I don't know that I have an answer to the question.
I did not. So I first found out that I was pregnant when I was touring Finland, and that like everyone was like, it was like sauna, right? It was like we were going touring saunas.
Yeah. You're a
And so,
sauna
of course,
VIP. Yeah.
I was, and I'm like, holy crap! Like, what am I gonna do? So I felt like at that early stage, I mean, literally, like I found out I was pregnant in Finland. So I did do a lot of sauna touring and stuff, and cold, cold plunging and all that at that stage. But right after, when I got on the plane, come back home, sauna was out. And the reason I sort of erred on the side of caution, I mean, I talked to women in Finland and they were like, oh yeah, I sauna throughout pregnancy. And like, you'll find those anecdotes, right. And it, certainly in that culture. But there is a body of evidence, mostly looking at like hot tub, and this is common, like it's common knowledge, like pregnant women shouldn't get in the hot tub, right? I mean, like you go to any spa, like it's like known, but there's a body of evidence that it can, something might increase the risk of like sort of like a fetal alcohol syndrome, sort of thing in offspring, where it's like, or even neural tube defects. So I was concerned that going in the sauna perhaps could increase the risk of something like that. And so I decided that it just wasn't worth it. And so I did not sauna at all, like throughout pregnancy. And I even waited a little bit, like while breastfeeding and stuff. Like I waited probably like 6 months or so before I really got back into sauning. Now, all the while I was exercising throughout pregnancy, and so many benefits to that. But kind of back to your question, the other protocol I do is at night. And it's interesting because doing the hot tub at night, I'm like, we're in that hot tub, and it's kind of also a time that my husband and I get together away from our child, right? I mean, it's like our time. Yep. We're like out, the stars, like dark sky, like it's nice. And that is something that like my husband likes to do it, like literally, like he wants to do it every night because it helps his sleep so tremendously. I don't have as much of an issue with like my latency or my sleep, like in general. But he does, like he falls asleep, like my natural bed, like I'm asleep by 9:30, and it's like, no, like I can fall asleep within like, I'm asleep in 10 minutes. Like I get in the bed and like I can be asleep in 10 minutes. Like he is not that way. And so doing the hot tub, and he likes the hot tub, that really helps his sleep. So I end up doing that a lot as well. And sometimes I'll do like both. I'll do the sauna and hot tub in the same day. It all depends. But exercise is the most important. I mean, like that's like that I have to get. And I go for the vigorous type of exercise. There are studies looking at intensity with respect to dementia risk, cognitive, impairment. You'll find all sorts of things, but like I think the most common thing that is pretty thematic is that the more effort you put in, the more time you put in, the bigger the benefit with respect to cognitive health. So dementia risk, and also it depends on how it's. I'll give you an example. There was a longitudinal study where women, by the way, as you know, are, I think, proximately at a twofold higher risk for Alzheimer's disease than men.
That's right.
Super interesting. But so this was in women, and they were studied for decades, and I think it was like starting from like the 70s up until like 2010 or something. And they came in for a physical, like they were put on a bike, exercise bike, and their like fitness was measured, like it was like empirical data. Okay, and this was like, I don't know, 5 to 7 times. So like over the course of it was like
40 years or something.
Yeah, exactly, something like that. And the women that were the most fit by their measurements on the this like cycle test they did.
Yeah, it was probably VO2 max.
VO2 max, and I think so it was cardiorespiratory fitness. That's right. And then those women that were the most fit had they were like the reduction in like Alzheimer's like risk was like so robust. I think there was something like they were 9 times less likely to get, or something crazy like that. The ones that were moderate, like, so they had like a moderate cardiorespiratory fitness, they were like 4 or 5 times, like they had a 4 or 5-fold reduction. But then you'll like look at another prospective study, same deal where they don't come in and get anything measured, but they come in for a questionnaire, or they get a questionnaire every, whatever it was over the course of like 40 or 50 years or something. And this like so they answer all these questions like, oh, how often do you jog or bike, or do you play tennis or whatever? And you look at that study and there's like no association between physical activity and dementia risk. And I'm like, like that's interesting, because this other study where they're actually measuring something showed a robust reduction in dementia risk. And so it hits home this like, okay, what study are we looking at? Like you'll find questionnaire studies that also show, a benefit, like, people that are physically fit, and the more fit they are, there's like a linear dose response effect, right? Where you see, people that put in more effort, they're training for a longer period of time, and they're, doing it at more vigorous or more volume, both, right? Like they have the greatest benefit with respect to dementia risk, which isn't so surprising to me.
Yeah, but the studies that are unambiguous, as you said, are the ones that actually measure VO2 max, because there's no denying what you're measuring. It's a very objective measurement, and it basically takes out the training component because it captures that benefit. It's the readout state of the training. And it basically says, look, maybe it doesn't matter if you do 5 high-intensity workouts a week or 2 high-intensity, 5 low-intensity. Like, what matters maybe more is the output. I don't know if that's the case, but there's no denying that people who have a high VO2 max are doing something that people who have a low VO2 max are not. And that's what's being captured in those studies. And, the numbers are astronomical. I won't go into them again because people on this podcast have heard it too many times, because I can't stop talking about the benefits of having a high VO2 max. But I want to touch on something else you just said a second ago, which is you noted that women are indeed at twice the risk of Alzheimer's disease to men. Of course, Parkinson's flips that. Men are at higher risk. But focusing on Alzheimer's disease for a second, is there any evidence that there are gender differences in response to exercise? In other words, are women more responsive to the benefits, or more amenable to the benefits of exercise than men because they are at a higher risk genetically?
I haven't seen the studies looking at the response to exercise with respect to the sex differences, but like, as you mentioned, like there's definitely differences with respect to their, Alzheimer's disease risk. So, and I do know like there are different mechanisms that could, so women have different, like there's different metabolic responses to exercise, maybe also, hormonally different. I'm just like, this would make sense, right? Like, I don't know that this has all been studied. I haven't seen that data, but like hormonally different responses to exercise, that would be plausible. Immune system effects as well. There's so many. Exercise is affecting the immune system. So we haven't even talked about like myokines. Like these are like, these are molecules being secreted by our muscles. We talked about lactate. That's not a myokine. That's it's a metabolite. But I mean, physical activity, when we force our muscles to work hard, we're making something called a myokine. Sometimes it's referred to as an exerkine, but like this is irisin is one, IL-6 is another. There's other ones as well, but like these are also affecting the brain, and they're affecting cancer risk, right? There may be differences in respect to like myokines that are being secreted with respect to how the stress of exercise, how that's response is happening.
Let's dive into that. Let's talk about cancer, because Rhonda, while I think both of us are, and I think there are others who share this point of view, completely convinced, in fact, I just don't see how one could not be at this point convinced of the benefit that exercise poses to the brain. It seems much harder to make the case for cancer. In fact, when you think about some of the things that are such obvious problems with respect to dementia, for example, disrupted sleep, poor exercise, etc. Clear relationship. Very hard, at least for me, to make the case that bad sleep is related to cancer, although I think it is, right? But I don't, the data aren't clear, right? Like you can certainly make the case that horrible sleep would lead to a weakened immune system, especially the cellular system, more than the humoral system, would easily lead to an increase in not necessarily cancer initiation, but cancer propagation. But again, the data are so much less obvious. Let's talk about this relationship between exercise and cancer, right? On the surface, it should make sense. Exercise is good, cancer is bad, more exercise should mean less cancer. How compelling are the data? And I'll admit that I haven't gone as deep here as I have on cardiovascular disease and neurodegenerative disease.
So it's also another area that I'm very, very interested in because, I mean, as you start to get into your fourth decade of life, you've now had a friend or a family member that has come down with cancer, and you see just, I mean, you're a physician, so of course you've experienced it on, at a different level, but like you just see how terrible it is, to get cancer, and that it really the best hope is obviously to try to not get it, to prevent. And there are, as you mentioned, there are things that, there are definitely things that can modulate that risk that are a little, genetic, wise, that are harder to kind of move the needle. But overall, so with respect to cancer incidence, it's interesting if you look at like, you were talking about some of these elite athletes, right? I mean, like people winning the Tour de France and people that are Olympic medalists, or maybe that have even just entered the Olympics. I mean, you have to be quite an athlete to just get into the Olympics, right? And there's been a lot of interesting studies, quite a few that I have seen, and these are studies where observational data again. So I'll be obviously caveated with that. You're looking at people that have just entered the Olympics, and over the course of like from 1912 to 2010 or something like that, like just decades. And looked at all-cause mortality, cancer-related mortality, and compared it to like the general population. So there's a couple of studies that have come out of the U.S. And if you look at both of those studies, one of them was actually looking at medalists, and the other one was just looking at people that like entered the Olympics. They saved about 1.5 to 2 years. Like in other words, they were of life from not getting cancer, and about 5 to 6 of just basically they had a 5 to 6 year what you could call lifespan extension compared to the general population. Same with like French Olympians as well. Some very similar, where it was like 5 years. They lived on average 5 years longer than people than the general population, and it was attributed that they had basically saved 2 years of life from not getting and dying from cancer. I guess I should say dying from cancer, because they are two different things. But that would be like at the elite level. And it's interesting because you go, well, 2 years, like that's it? I mean, that's how I see it. I'm like, really? Like 2 years? And it's funny because I remember when I was a postdoc, my postdoctoral mentor, Bruce Ames, he had said to me once, or actually more than once, I once read, there's, of all the things that you can do, like if you prevent cancer, you really only save about 2 years of your life. And I always thought, I'm like, no way. But anyways, so that would be like at the extreme end when you're looking at the actual athletes. They're definitely less likely to die from cancer than general population people. But when you're talking about prevention, so there's a difference between, if you read a study and it says, people that are physically active are X% less likely to die from cancer, so cancer mortality is decreased. That's not necessarily the same thing as not getting cancer, right? That just means you're not dying from cancer. So the studies looking at cancer prevention really seem to focus on a specific type of exercise, and that is aerobic exercise. For whatever reason, there's not a lot of literature on strength training and cancer prevention. You can find studies on strength training and cancer-related mortality, but with prevention, I really it's sort of focused on, for whatever reason, on aerobic exercise, and it does seem like There are certain types of cancer that are more responsive to exercise with respect to, basically having a reduced risk. And some of those cancers are ones that are we should care about. So breast cancer, what's the lifetime risk of breast cancer? For a woman, it's about 1 in 8. It's pretty high for the average woman, right? Of course, many different lifestyle factors play into that, and exercise, is one of those factors. Colon cancer is another one that seems to be quite responsive. Lifetime risk of colon cancer for average woman is like 1 in 23. For a man, it's like 1 in 25 or something like that. The reason I'm mentioning, as you know, Peter, lifetime risk of cancer, like if you're talking about like esophageal cancer, some cancer where it's like 1 in 500, I mean, you're more likely to die in a car wreck than get esophageal, like, one of those cancers. I think it was esophageal cancer, but you get my point, where the lifetime risk is already kind of, quite low for the general person or the average person. So breast cancer, colon cancer, and then there's a few other cancer types that are quite responsive, but those two in particular kind of stand out because with prevention, and also with respect to people that are diagnosed with cancer and have those cancers, and then they engage in physical activity as well. It's very, you see a very robust response with respect to reducing cancer mortality and also recurrence being, it's like 50%. So you see cancer mortality is reduced by 50%, cancer recurrence is reduced by 50% in those individuals diagnosed with breast or colon cancer or colorectal cancer that are engaging in more physical activity. So the question is, well, how much? And you mentioned you have a lot more knowledge with respect to cardiovascular disease. And I would argue the data really, I would say, suggests that you actually probably need to do more exercise to sort of reap the cancer preventative benefits than you do cardiovascular benefits, or, even, some of the metabolic benefits. And I don't know why that is, but it seems as though, like getting more to that upper limit of what these, committees are recommending. So 300 minutes a week of moderate intensity exercise, or maybe 150 minutes more of what they would define as vigorous, which actually I think is their vigorous is a little bit below what my definition would be. But anyway, so it seems like the amount of exercise, you actually have to put in a little bit more time and effort for the cancer. But any amount is beneficial. So it's not like, oh, well, I can't do 300 minutes, therefore I shouldn't even care. Well, that's not true, because there are benefits, even with any type of physical activity. So, that's all the observational data. And you can find anywhere between a 10 to 20% reduction in basically people are that, they're less likely to get breast or colorectal cancer, 10 to 20%. Again, when you're talking about a type of cancer with a higher lifetime risk, it makes it's more compelling, right? So it's always
I'd be curious to see if the data line up with the cancers that are known to increase in risk due to obesity. So right after smoking, obesity is obviously the second leading modifiable risk factor associated with cancer. I've always thought that was an oversimplification because we use obesity as a proxy, but I think it's probably insulin resistance that's the true marker that obesity is serving as a poor man's version of. It would be interesting to see, because there are certain cancers, including breast and colorectal, by the way, where obesity amplifies risk. There are other cancers where obesity doesn't seem to play as much of a role. It would be very interesting to align the exercise data with the obesity/insulin resistance data and see if exercise is disproportionately reducing risk in those cancers for which obesity is a risk.
Such a good point, Peter. And I think there is at least some data to suggest that you are correct with that. So, what is there, 13 or so cancer types that obesity is known to
Yeah, it's either
the
13 or
risk for?
17, something like that. Yeah.
Yeah. And breast and colorectal cancer are on that list. And so, yes. So the aerobic exercise, I think there's direct mechanisms. So aerobic exercise is, directly you're making those myokines, and some of these myokines have been shown to to basically decrease the production of like growth factors secreted from cancer cells, and they also like kill, are killing cancer cells, through a variety of other mechanisms. Also, the anti-inflammatory effect, from exercise as well. So you're having a strong anti-inflammatory response, but also like there is the little bit of that. Okay, well, exercise is also like improving insulin sensitivity, and it's you're in combination, particularly in combination with dietary strategies, weight loss. The weight loss itself is basically a important component of the cancer, reduced cancer risk. So I think I think it's a combination of these things where it's like the direct effects from exercise, and there's also, it's really interesting because, as I mentioned, people that even have cancer, the response, like it seems like, physical activity, like I am not an oncologist, and many more oncologists than I do. I don't know that I don't know how common it is for oncologists to prescribe exercise as an adjunct sort of treatment to, whatever the person, whatever type of treatment, whether it's immunotherapy or radiation or chemotherapy or a combination, whatever. I don't know how common it is, but the data is more and more compelling. And I think it's become more and more compelling over the years that really exercising seems to be very important for reducing cancer metastasis and also dramatically decreasing cancer recurrence. And so a really interesting mechanism by which this is likely occurring is literally through that shear force mechanism I was describing for the brain. Well, as cancer cells, tumor cells sort of escape the site of the tumor and they make their way into circulation. It's called a circulating tumor cell or a circulating cancer cell, depending on the study you read. And, these circulating tumor cells are like traveling throughout the vascular system to distant sites, and they sort of take camp, and then like it's like kind of the seed of, a new tumor forming in another tissue. Well, it's really interesting because these cancer cells are so messed up, as like they're completely wonky and very, very sensitive to stress, any type of stress. They have these mechanoreceptors on their cell surface that are responsive to force, shear force. So when you get your blood pumping, it's like a hurricane that like just wipes it out. They die because they can't stand just the sheer force of the blood flow through, the vascular system. And so, and this is where you'll see, studies, you can sort of pair it. So you can pair the mechanistic studies, and there have been some studies looking at circulating cancer cells, and it's like people with those are like 3 times more likely to have cancer metastasis and so on. But there are studies showing that like physical activity like dramatically decreases, and there's been randomized trials showing it dramatically decreases circulating cancer cells in people compared to whatever the other standard treatment that they're being given. And so like kind of pairing that data with looking at other data where exercise is being prescribed to patients, and it is beneficial with respect to their cancer metastasis reduction and also mortality reduction, like 50% mortality reduction, versus recurrence as well. So I do think there is substantial evidence to suggest that being physically active is a good measure for cancer prevention. And, again, there's also a lot of differences. There are sex differences as well. Like, I don't know why, but in some cases women respond better, and there's certain cancer types that respond better. Lots of variables here. Like I feel like I'm speaking in a general way, but there are lots of things to consider, right? There are cancer types and there are sex effects, and there are, as you mentioned, other covariates. There's obesity, and there's, insulin resistance and age as well. So, I mean, there are lots of, it's nuanced as usual, but I do think that you can make the case that what can I do in my life to reduce my risk of getting cancer, reduce my risk of dying from cancer, reduce my risk of getting Alzheimer's disease, reduce my risk from getting dementia, reduce my risk from getting cardiovascular disease, reduce my risk for type 2 diabetes. Like, the only panacea there is exercise. It's exercise, right? I mean, it is the case. And unfortunately, it's the thing that you have to put the most effort in. It's certainly a lot easier to take a supplement, to take a pill. I do think there is an argument that omega-3 is one of it's up there, I think. I think getting yourself to a good omega-3 status and defining what that is still like being investigated. But I do think that's a low-hanging fruit that should not be ignored. But exercise, as you've talked about many of times, is the king. It's the king. And that's the thing that you should focus on. Obviously, if you're obese, weight loss, exercise is part of that program. And like I don't think that anyone that's obese should be worrying about all the other things. Like, they need to lose weight, and any personal trainer and coach probably is going to help you do that. Just you eat less. That's calories in, calories out. It matters to some degree. If you're not eating as much.
Yeah, but as you said, exercise matters not just on the energy balance side, but exercise makes you, for example, more sensitive to satiety hormones. So, look, I have kind of a belief here that the person who is overweight, the person who is obese and who is clearly eating more than they should be, isn't doing that by choice. Maybe some are, but for the most part, it's hard for me to imagine there's someone who's listening to this who's obese, who isn't wanting to not be obese, and who is otherwise struggling with hunger, right? And I think that, that's one of the challenges is, why is it that a person who is not in energy balance is not responding to the normal satiety signals? And I think there's a lot of reasons. On the food science side, we could talk about a whole bunch of reasons why our food has been hijacked. Our food is void of nutrients. Our food is hyperpalatable. It's far too available. There's a whole bunch of reasons. But I think one thing that doesn't get enough attention is this thing, which is an exercising person has a better sense of nutrient requirement. They have their body physiologically is more in tune with their appetitive needs. And so even though I don't think exercise matters as much as intake purely on the energy balance side, in other words, I think it's more about reducing input than increasing output, a part of that equation is the feedback loop that exercise brings. So yes, exercise just matters. And I also think that, especially in this discussion of cancer and breast cancer as the example you brought up, so many women are so petrified of hormone replacement therapy because of this awful, study, the Women's Health Initiative, which was completely misinterpreted. But just to use one example of what we spoke about, even the people who ran the study, who to this day, some of them, at least a subset, still maintain that conjugated equine estrogen plus MPA, the synthetic progesterone, increased the risk of breast cancer, even those people will acknowledge it did not increase breast cancer mortality. So even if you take the most favorable to the WHI, the Women's Health Initiative study, reading, the reading is that conjugated equine estrogen plus MPA increased the incidence of breast cancer by 0.1% in absolute risk, but did not increase breast cancer mortality. So here you have basically a non-event that has most people panicked senseless, most women panicked senseless when confronted with taking hormones during the perimenopausal period. And yet at the other end of that spectrum, we have a treatment that has more than a log-fold benefit in the other direction, i.e. in reducing risk. Right. And I wish people would just allow their attention to be allocated proportionate to the size of the impact.
I'm 100% with you. And with to kind of even just highlight or emphasize what you just said, there are studies with women who are doing moderate drinking, which, depending on the study you read, it's for women, moderate drinking is it's 2-3 drinks a day or something. It's a lot.
Wow.
And like that literally like translates to a lifetime risk of breast cancer. It's like 1 in 6 or something like that, where it's pretty significant, but you don't hear about women petrified of like drinking, you know, like 2 glasses of wine a night, which, I mean, some people do. Like it's, you know, it's actually not uncommon. And so it's like, again, it's one of those things that you were saying where it's like looking at like what's going to impact my risk more, what is going to lower my risk more, like what should I focus on? Like what's like the most important thing? I think obesity does, you know, it absolutely impacts breast cancer. Same with, you know, physical activity in the opposite direction has it. I mean, it's just really enormous benefits. And then alcohol consumption is another one. Even a mild alcohol consumption. I would say that like, I don't want to go there because it's like it's complicated, and I can't even like begin. Like there's
I've
so
gone
much.
there. I've gone there, Rhonda. I've gone there. I go out on the limb, and I'm going to say it. There is no amount of alcohol that is healthy. The J curve is a misnomer, and what I think I would say is somewhere between 0 and 1. There's probably there's not that much of an increase in risk, but there's not a reduction in risk. Right. So
For what?
For mortality in general.
Mortality.
Yeah. So in other words, you know, they talk about this sort of J curve where complete abstinence is a greater risk than, you know, 1 drink a day. But I think both the Mendelian randomization makes it clear that that's not true. And then secondly, when you look at all the confounders of the people who are drinking 0 drinks and what confounds their mortality, I feel very comfortable saying that there is no dose of alcohol that is healthy, but, you know, at a very low dose, again, probably 4 to 7 drinks per week, it probably has immeasurable, not immeasurable. You probably can't quite quantify the harm. Mm-hmm. That would be my take. And so I'm comfortable saying that. I really feel confident that that is the case. And that, you know, things like the French paradox have far better explanations as one example.
Yeah, I tend to think, I mean, the data is also a mess, I agree with you. And it does seem like, you know, like, can you have your weekend, you know, glasses of wine, like Friday,
absolutely.
Saturday? I think you can. And, you know, with respect to the cancer risk, and like that's considered, you know, it's mild. I mean, it's less than, you're having less than 1 drink a day. And the only evidence I've really seen against the mild is on the National Cancer Institute site, where it's one of those cancers where it's like 1 in 500. Like it increases your risk of a cancer that you already have a lifetime risk of 1 in 500, and it's still less than 1% of an increase. Like to me, it's like your lifetime risk.
You can't measure it.
You can't you can't measure it. Yeah,
it's a classic example of the dose makes the poison, but don't confuse that the poison is a poison, right? So another example would be cigarettes. If you smoked a cigarette twice a week, literally 1 cigarette twice a week, would your risk of cancer go up? Yes, but you wouldn't be able to measure it. That doesn't change the fact that cigarettes are harmful, right?
Not to mention heart disease, like that. That is not a linear like that. Like
No. I mean, but even just focusing on cancer, right? Like, it really comes down to kind of establishing causality, right? Is tobacco causally related to disease? Yes, it's a harmful thing to take, but the dose matters, right? Like, again, just being glib, 1 cigarette a week it's probably increasing risk, but we don't live long enough to see the separation of those Kaplan-Meier curves, right? Maybe if our natural lifespan was 500, 1 cigarette a week would be sufficient to see a spreading of those lines. But at an 80-year lifespan, you have to get up to 10 cigarettes a day before we can see where that is. By the way, I'm making that up. I'm not advocating that one can smoke up to 9 cigarettes a day, but you know what I'm getting at, right? And I think that's my point. With alcohol, it's simply just a question of that. But I just want to make sure people aren't taking away from this that, you know, look, I probably have anywhere from 0 to 4 drinks a week, but when I'm drinking those 4 drinks across 2 or 3 days, it's not going through my mind that this is healthy. It's like, this is a hedonic pleasure that's not good for me, but it's enjoyable.
That's enjoyable. How do you feel about ApoE4 carriers and alcohol consumption. Like
our view in the
2 to 4.
practice is that they are indeed more susceptible to the deleterious effects of alcohol. And also, I would say they're just more susceptible in general to the deleterious effects of poor sleep, which is one of the ways that I think alcohol is disproportionately hurting the brain. I think poor sleep is causally driving Alzheimer's risk and cardiovascular disease risk. I'm less clear on cancer, but in as much as most people that are drinking alcohol are doing so in the evening, and anybody who's used a sleep tracker, you don't need to be Matt Walker to very quickly do the experiment on yourself and compare a night of sleep with no alcohol, a night of sleep with alcohol. They're different. And so through that lens, I would just say, if we have lots of patients with E4 in our practice, including a number of E4/E4s. We even though those patients represent only 2% of the population, they're probably about 7 or 8% of our patient population. And again, we say, look, unless this really means the world to you, it's probably not worth the drink. And if you are going to have a drink, here are some principles for how you might minimize the damage, right? In terms of the number you might have, how long you might have it before bed, that kind of thing.
Exercise. When I, a couple of things, just because you brought up the sleep. And for one, so I started wearing a continuous glucose monitor largely because of you. And
I've been a very bad influence on some things you've done. The lactate monitor, the CGM.
Yeah, well, I started wearing it when I was a new mom. So this was, like 5 years ago. And
that can't be a good time to wear a CGM, but although it must have been interesting, right?
It was extreme. So here I am coming into this like, I'm going to learn about the foods I eat and how my body, responds to those foods. And lo and behold, the biggest and most compelling and most important data point, or many points, because I had many of them, that I sort of learned from wearing my continuous glucose monitor, which I'm sure most of your listeners know about, where you're measuring your glucose level like continuously, was the effect that my sleep interruption had on both my fasting blood glucose and my postprandial, where I was I could get what can be considered like pre-diabetic. Like I was like blown away. I was like, this is insane. And then what I also sort of gleamed from this was that when I was on days, and the effect lasted at about 48 hours or so. When I did work out, and at the time I was doing a lot of high intensity interval training. It was like an hour-long spin class I used to go to, where they do all this interval training. That it almost completely blunted that effect, where even though I was dog tired, last thing I want to do was go to my damn spin class. I was just like, this is like, I can't, it's going to be bad for me if I go. That's how I felt. Like it's going to be bad for me. But it was completely the opposite, where this crazy glucose, dysregulation and whatever the cause is for that. I'm sure you know much more about that than I do. It was almost completely blunted, and it was so profound. And it was like all the food stuff, I learned a little bit of interesting, but really that was the thing that for me was like, I have to work out no matter what. Like I have to, no matter how I feel, it doesn't matter. It's beneficial. And there's also, it was a really interesting study, caveat, observational data. You never know, like there's reverse causation, all the problems with any study, but it was looking at the, sleep habits. And so people that had slept, was poor sleep or interrupted sleep, something of that nature. I can't remember all the exact things that were measured with respect to sleep, but people that, didn't sleep as long or had poor sleep, whatever, by whatever measurements, had a higher all-cause mortality, which is not that surprising, but only in people that weren't physically active. And to me, I was like, oh, wow, that's like,
interesting.
It's interesting. And it's like, exercise can forgive a lot of sins in many ways. It really can. And, so you're talking about your patients with E4, and I'm sitting here going, oh my gosh, I've got one of those. And I'm already like, it's a lot, it's a burden. You have to calculate things. You have to be very, specific in the actions you take, and like things that you don't do, things you do. Right. And for me, it's like, okay, am I going to I'm having a party, we're going to have some mimosas, whatever. Like, I'm going to exercise no matter what. Like, and that might take off some of the stress, right? I'm not saying I go out. Like, I personally, I hardly drink, and mostly because I am E4. I have one allele, and I have pretty much come to the conclusion that my brain can't repair damage as well as my husband's, who doesn't have an E4 allele. With that said, I occasionally I'll have like maybe, a glass or two of wine. Usually that's my preference, but, a week. But even, last time I had a drink was like Valentine's Day, so it's been a while. But at least I think with the exercise, again, it's like, it seems to forgive a lot of sins, honestly.
I like that way of describing it. Yeah. I think,
I kind of borrowed that from Stuart Phillips. I think he was the one that said it to me, because I was like, this is like,
yeah, that's right. I heard that. That's right. I remember him saying that on your
Stuart
podcast.
Phillips. So he gets all the credit. And that of course brings us into the whole like protein intake and muscle mass world.
So let's set this up for listeners, because you and I have talked about this a lot. In fact, I remember probably the last time, this is pre-COVID, because we were both in San Diego. You and your husband were over for dinner. I think I cooked up some fresh venison that I had just killed, and we were kind of talking about protein. And I think at the time, both of us were kind of struggling with two competing ideas in geroscience. And those two ideas that seemed dialectical, they seemed at odds, was on the one hand, there's this body of mostly kind of animal literature that suggests lower protein intake is associated with a longer life. But on the other hand, there's this literature that says lower protein intake is associated with more frailty in humans, and that's associated with a shorter life. So how do we reconcile these two things? And we didn't have a great resolution on that. We both kind of felt like we were sort of scratching our heads, thinking, at least, I don't want to speak for you, but my thinking at the time, this is again 3.5 years ago probably, was we just got to find the minimum effective dose. What's the minimum effective dose of protein to not undergo mandatory catabolism? And that's what the dose is. So, and of course, it's not clear how you find that dose. Theoretically, you would use a metabolic cart and try to identify nitrogen balance and things like that. But of course, no one can do that outside of a lab. So it was a bit of a head scratcher. Now, my thinking has evolved so much on this, but I'd like to hear, first of all, I'd like to hear your formulation of the problem. Did I formulate it the way you would? And I guess more importantly, tell me how you're thinking today.
Yeah, so you actually did a very good job formulating sort of my mentality with respect to protein intake and longevity back in 2019. Prior to that, but like even up until not even that long ago, to be honest, there's like, as you mentioned, a large body of animal evidence, but also like there was coupled epidemiological data where people are looking at, vegetarians are taking in lower amounts of protein and they're all-cause mortality and their cancer mortality. And there's just study after study after study. And they have lower all-cause mortality, lower cancer mortality, but only in those individuals who are not obese, not sedentary, are sedentary, are smoking, or, they had some unhealthy lifestyle factor. So in other words, the people taking in higher protein, animal protein, who were basically healthy, had a similar cancer-related mortality, all-cause mortality, as these vegetarians. Okay, so
and similarly, studies where they normalized for fruit and vegetable intake, high protein versus low protein, no difference in early mortality, right? So yes, it seemed that a lot of the data that were espousing low protein were confounded by lifestyle choices, and high protein was also negatively confounded by high calorie as the most obvious.
Exactly. And with the animal data, and this is probably where my mentality has shifted the most, because I wasn't really of the opinion that vegetarian diets were superior to, meat eating, or I guess omnivore types of diets that were healthy omnivore types of diets. Like I like, because of what you were saying, like that data was, it was nuanced, and it wasn't just like, so that wasn't something that has shifted in my, the way I've been thinking. But with respect to, a lot of the animal data and the, and the mechanisms, and you can, restrict a mouse of protein and make it live longer and not get cancer, and, all these things that you see study after study. I mean, it's just like the longevity science and that whole field is like dominated by that, like at least was. And there's now, I think, some pushback going on, but there's still a large, group of scientists that are, still, publishing lots of animal data. And this is where I sort of started to look into some of these exercise physiologists, people that like Stuart Phillips. I know you've had like Don Layman on, yeah. Like exactly, like these like giants in the field that are doing the research. And Brad Schoenfeld is another one where they're looking at protein intake, they're looking at strength training and its effect in humans, right? In humans, on muscle protein synthesis, and also just like looking at data with respect to muscle mass and, all-cause mortality and, Alzheimer's disease, dementia. We didn't, talk about that, but like strength training also, can modify that risk. Also cancer mortality. And so,
strong grip versus weak grip, monotonic change in grip strength, 70% reduction in incidence and mortality from dementia. I mean, 70% reduction in risk. Remember, people don't understand, you can't reduce risk more than 100%. So it's not like increasing risk, which can be 100, 200, 300. When you're talking risk reduction, 70% is staggering.
It is.
Strength matters. Yeah.
It does. It does. And you're not like there, as you know, and many of your listeners, like there's two important signals for, your muscle. Strength, obviously a big component of that is like physically working them. But, protein intake plays a role there as well. And I think it was Stuart Phillips like phrased it this way where, the animals that are being studied in these labs are in a sterile environment, right? They're in a sterile environment. They're not being exposed to influenza and all these infectious diseases. And, like any of us we've had a parent or a relative or someone that has gone into the hospital and maybe had bed rest, and then come out and like I had a grandparent who literally couldn't walk after a back surgery. It was like, for forever. Like that was it. That was like, that was their downfall. It was like the trajectory just went down, completely down. And, so losing that muscle mass, when you're in older age, obviously building up a bigger reserve in youth, like in middle adulthood, whatever, like that's very important. But like, these animals that like, people are manipulating the protein restriction in them, like they're not being exposed to that, right? They're not losing like, I don't know what percentage, like it's pretty intense, like how much you can lose from like 3 weeks of bed rest. And also like, as it's been pointed out by people on your pod, I think Matt Kaberlein, like these, mice are dying from like cancer, right? Like cardiovascular disease. They're not dying from the same diseases. And it's not even the same type of cancer that humans get. Like right. We get a lot of these epithelial, like tumors, solid tumors. They're like dying from lymphomas, right. And, like that's, so there's a lot of differences there as well. Where I don't like, there's a lot of interesting, and I'm the one I've talked a million times about animal studies. And I think they're important mechanistic data. Like there's things you just won't ever get from humans, but at the end of the day, I started to realize that, looking at this, mice in a sterile environment where they're not really being exposed to the same stimuli as humans, things are very different in terms of aging. I was like this, it was falling apart in my mind, basically. It was like all falling apart. I'm like, oh my goodness, this doesn't make any sense. Like this isn't what to be looking at if I'm wanting to really focus on healthy aging for myself, and for for everyone else, right? So I think that's kind of what was the tipping point for me, was just kind of that realization of the importance of muscle mass, and how, some of these animal studies, you look and you're like, oh, they have, they're a little bit of an improvement in their cardiovascular health. And I'm like, exercise. Okay. Is that better than exercise? No. Like the things that were improved, I was like, exercise does that. Exercise does that. Like, this isn't convincing me that I need to like do that, So, I think it was just kind of like the shift in the way I viewed the data, like the lens I was seeing it through.
Yeah, I'm still waiting for somebody to demonstrate for me that if there is an increase in the risk of cancer associated with higher protein intake, in humans, I'd like to see that quantified. But I would like to see somebody demonstrate that if there is an increase in risk, that quantifiable increase in risk is greater than the offset of sarcopenia. Because that's something for which there is no ambiguity. We have all the data in the world to point to the devastation of sarcopenia on an aging population. And we know full well that two things have to be true to avoid sarcopenia. Adequate protein intake, which as you age gets bigger and bigger. That number goes up and up and up due to anabolic resistance, coupled with strength training. So here we have something for which there is no uncertainty. You must consume increasing amounts of protein, and you must do strength training to ward off sarcopenia as you age. And if you don't, here's your mortality trajectory, and it's awful. Let's compare all of that to this questionable risk for which, frankly, I don't see data. And I'll add one more point to what you said, Rhonda, which is I think this story got confounded by our good friend C. elegans, right? So let's go back 29 years, roughly, call it 30 years directionally, when some very seminal and interesting work was published looking at the DAF16 mutation in, or maybe it was DAF2. I can't remember if it was DAF2 first or DAF16, but it was the analog of the IGF receptor. And if you knocked out that gene, you could double the lifespan from roughly 2 weeks to 4 weeks, or 4 weeks to 8 weeks, I forget what it was, of C. elegans, this worm. And the implication of that was profound. I mean, I don't want to downplay the most important takeaway from that, which was lifespan was malleable. That turned out to be very interesting. I could go on my rant about why C. elegans is not an organism or an animal model that offers any insight into us, based on its cell, based on a whole bunch of things about its biology. But nevertheless, it somehow became knocking out DAF2 or DAF16 was tantamount to dropping IGF, insulin-like growth factor 1, to 0, is the key to longevity. And the way to do that is to have no protein. And I think that story is so incorrect, but somehow it's become part of dogma. So I think that's the other piece of this that just kind of won't go away.
It's funny because I've done those experiments with my own hands when I was at the Salk Institute, and I was in Andrew Dillon's lab, who had trained with Cynthia Kenyon, who made the discovery back in, was it early 90s or something?
Yeah, I feel like it was 93 or 94.
Yeah. And, it was very exciting for me at the time, because it was like, oh, this is a homologous gene we have. And I'm watching it go from a 15-day lifespan to like a 30+ day. And not only that,
the health span was remarkable. Yeah.
They were youthful. I mean, you could see them, you look at a microscope and you see how they move around. It is very apparent they were acting like a youthful young larva that had like not been born long ago. Right. But then you also realize they go into this dour state where, like, in order to get that, lifespan extension, they're like going into this like metabolic stasis, and like this thing that we don't do, like humans don't do doubt. Like we're not, it's a completely separate sort of pathway that is required for that lifespan extension. And I think, to your point about the IGF-1, the insulin like growth factor receptor, and also the insulin pathway. They're kind of both like tied into that. The fact of the matter is that is a growth factor. And, as we have talked about, you and I have talked about this before, growth factors in the context of a tumor can allow tumor cells to override cell death mechanisms. So they can continue to survive when they otherwise might have been, signaled to die, right? And so there can be a problem with too much IGF-1 in the context of a tumor, and what causes that high IGF-1 is up for debate. But I think at the end of the day, it's not that high IGF-1 that is necessarily causing the tumor. I think, like there are things that you can do in your lifestyle, like exercise actually causes IGF-1 to go into muscle, where you're repairing damaged muscle. It's helping muscle repair. It goes into your brain. It's important for like neurogenesis. A little bit of controversy there, I know, but like I am in the camp that you adults are, there's multiple studies showing that you can take an older adult, train them for a year, and their hippocampus will grow by like 1% to 2%. Like there's multiple studies. Also another study showing this with the subventricular zone. So these are two regions of the brain where it is my opinion, I believe data that says that adult neurogenesis, or the growth of new neurons as an adult, is occurring. And I think exercise is big.
And do you think that part of the vehicle for exercise to do that is through IGF-1?
Yeah, absolutely. It is.
Interesting.
Animal studies have shown that. Again, we all know about the caveats. It does it translate to humans? We don't know. But, oftentimes you have to take the whole body of evidence, the human evidence, couple it with mechanistic data with animals, and try to kind of put together a story to the best of your ability. That's all we're going to get, And so, IGF, like you always hear about, I want to lower IGF-1, but it's actually like important for the brain and important for muscle. And the way to get it to the brain is through exercise. Like that's known, and it's been shown again in human studies as well. So I think also part of the problem here is, in some respects, people that are doing, really impeccable animal research. Like you look at, the data and it's like, oh, they're doing this study. Great. Like this is a good study. Like you can't poke holes in it with respect to the animal world. But then sort of translating that to humans and considering, like, who are we talking to? Are we talking to an overweight, obese person? Maybe they're probably getting enough protein. They're getting enough, they're like, I don't know that they have to worry so much about protein intake. I think they need to focus on losing that unhealthy weight.
Yep.
But that's also really important. And I think some scientists and also health, and science communicators also sort of, maybe it's hard. You have to like, and I've been sort of guilty of that as well, like disentangling, who are we talking to? Are we talking to the obese person who clearly needs to focus on weight loss? Or are we talking to the healthy, physically active person who's now terrified to take protein in because they read about some animal study where too much protein increases mortality, right? And
also, I think age is such an important part of this again. So if you look at that, I think it was Levine in 2017 had that study where they look at the relationship between protein and IGF-1, but stratified by protein intake, low, medium, high. And then they looked at middle-aged people, so 50 to 65, and then people over 65. And in people aged 50 to 65, there was a relationship between protein intake and IGF. Higher protein intake was associated with higher IGF. Now, it wasn't a huge difference. This gets overstated constantly, but it was statistically significant. But what often gets ignored is the people over 65, there was no statistical difference whatsoever between protein intake and IGF-1. Again, this is taken as dogma that the more protein you have, the higher your IGF-1. And in people over 65, that's not the case. Now, why do I harp on that? I harp on that because it is in exactly that population that I am most concerned with sarcopenia. So if the message is somehow getting transmitted to somebody listening to this who's 65 or older, that I shouldn't be eating protein, And they might not even know that it's through IGF, but somehow high protein is going to give me cancer. And that's because someone who's telling them that is telling them that through the lens of IGF. The answer is, first of all, no, it's not. And secondly, the greatest risk you face, again, is going to be the results of low muscle mass and low strength. And even if we believed, which I don't, but even if we believed that in that age, in the younger people, eating more protein leads to more IGF, which is bad, I would argue that the absolute risk of death is so much lower in that group that the absolute difference in mortality between the younger and the older in the presence of high protein is no comparison. What I mean is, higher protein across the board is going to save more lives than it would ever hurt in younger people, even if you could convince yourself that higher protein intake was associated with increased mortality. And so, again, I find these data unassailable, especially in the older population. I think Matt Caberlin and I did talk about this on a podcast once. And I worry that, as you do, that information is not making its way clearly to people of the susceptible age group.
Right. And I think also that the thinking about a lot of people are focused on the recommended daily allowance of protein, right? Like, well, then, right. Like, what are these old ass studies that were not done correctly using the wrong tracers? What are they like, what is that telling me about how much protein I should take in? And this is also another sort of change in my, it was a turning point for me, because I knew nothing about the fault, the errors in how those
the recommended dietary analysis was determined.
No, I know everything about micronutrients and RDAs, but I knew nothing about the protein. And once I talked to Stu, I mean, it was clear. He was like, "Oh no, we repeated those studies, him and many others, using different tracers." And I can't tell you all the tracers and stuff because it's not my field. But it was like, no, we determined that the minimum was really more like 1.2 grams per kilogram body weight, not 0.8. And to me, I was like, oh wow, because you don't store protein. So it's like, okay, well, this is important because, I mean, that's a big difference. And then on top of that, when you start to get into the physically active people or elderly population, as you mentioned, anabolic resistance, where they're basically like their muscle isn't getting that signal, as well to increase muscle protein synthesis from the same amount of protein that their younger self would, right? So they actually need more of a dose, up to 1.6, 1.8.
We basically tell people, aim for 1 gram per pound, which would be 2.2 grams per kilo. Because the other thing that complicates it, Rhonda, is not all protein is created equal. So, if you're getting a reasonable amount of your protein from plants, you're getting a lower bioavailable amino acid. You're also not getting the same quantity of leucine, lysine, and methionine, which are probably the 3 most important amino acids anyway. So, one of the things Don Layman talked about was, if you really want to be rigorous about this, you probably want to track those amino acids. And you really want to say, look, make sure you're getting 1 gram at least of methionine per day, 2 to 4 grams per serving of leucine and lysine. Now, again, for a lot of people, that's too nerdy, but you can go through the math a couple of times with certain things that you eat repeatedly, and you'll realize that's probably more protein in aggregate than a person is used to eating. And as you said, when you start to factor in those 2 other categories of risk, right? More demand. So when you're doing those high intensity workouts, you are ripping apart muscle fibers. When you're lifting weights, when you're rucking, when you're doing all these other things we love to do, you're demanding more amino acids for the turnover. And then of course, anabolic resistance, I think, is the biggest issue. And something that truthfully, up until 2 years ago, I just wasn't paying enough attention to. I wasn't appreciating that my older patients had an additional problem that younger patients didn't have with respect to that signal.
So that's a lot, 2.2 grams per kilogram body weight. I struggle, for me to get 1.6, I'm supplementing, I'm taking whey protein. I just can't, it's like, how many meals?
Typically 4. Yeah, because it's
Yeah,
hard
you'd
to.
have to, it'd have
It's
to
hard.
be.
Yeah, it has to be. And one of like so for me, it's really 2 meals and 2 snacks, and the snacks are
Mhm.
just protein
Protein.
snacks. So
Right.
it's a shake. So one of them is just a whey protein shake, and then one of them is I eat these venison jerky sticks. So 5 venison jerky sticks is 50. They're 10 grams a piece, and they're really good. They're super pure venison. It's wild game, amazing product, and I should disclose I'm an investor in the company that makes them, by the way.
I was going to ask, do you make them? Is it like
No,
your
yeah,
own?
but
No.
I know the people so well. I know everything about it, and I know the quality is there. So those are 2 snacks, right? They're relatively low in calories, right? Like my whey protein shake doesn't really have anything else in it except some frozen berries and almond milk. And then the venison sticks are what they are. And then 2 meals that are, going to have protein. And for me, again, a lot of times like it's going to be an omelet and then, protein dinner. So yeah, it's not going to deny it. It's work. It's not, it probably consumes more of my dietary planning and dietary attention than anything else. I don't pay any attention to how many carbs and fat I eat anymore. I'm just paying attention to protein intake.
It's something I never really paid attention to much at all. Until, and I would say like last June or July is when I really started focusing on strength training, both for my muscle mass and also bone mineral density.
Yep.
Like that's another thing where it's like, you want a reserve of that as well, especially as a female, right? Like, so
Yep.
that those are like focusing on the strength training and also the protein intake. And it's been quite challenging. I've always sort of focused on micronutrients, and it is, it's still a focus of mine, and like making sure I'm getting enough of those. And I do supplement as well. In addition to trying to eat like leafy greens and getting some of the veggies and stuff, it's either going to be roasted veggies for me or like salad. But the protein intake is like, it's been challenging. And I find I typically do 3 meals. One of them is like a protein meal snack. So it's like some salmon or like a homemade turkey burger or something like that. But then the protein shakes also is where I just have to do. I guess that I don't really consider it a meal, but it kind of is. And it is like it's satiating. The protein like shake is definitely satiating, and it's even kind of hard because when you work out, like as you mentioned, your satiety, hormones go up. Like I'm not hungry. Like I don't like necessarily want to eat. Like it takes a while before I can actually like even get an appetite,
Yep.
So there's all these like competing things where I'm like trying to get the protein, but I'm like, I'm not really hungry. And I'm like, but I know I need it. And, so all these little important factors. And yet again, important to sort of, highlight that, I don't know that someone who is overweight or obese necessarily needs to focus so much on that, right? I mean, would you agree?
Right. That's right. Yeah. I mean, fortunately, most people who are what I call overnourished are also adequately muscled, and they can actually in the short run be okay losing lean mass. In fact, it's very difficult to lose heaping amounts of body fat while preserving lean mass. So we tend to focus more on the caloric restriction coupled with the training. We use the training as a way to offset some of that lean mass loss. And then we can come back to it. But yeah. Now that said, for people, so it depends on the strategy, right? Depends on the dietary strategy. So for people who are using tracking, kind of the caloric restriction way, we would still set a protein target that is at 2 grams per pound because of the satiating benefits that you said, right? Also, you have the thermogenic effect and the benefits of protein over fat and carbohydrate from a thermogenesis standpoint. But when you have people that are going about it via dietary restriction or time restriction as their strategy for cutting calories, it can become a little overwhelming. And when you force high protein, you sometimes end up getting high calorie with it. So that's where we would say, just don't pay attention to it as much. Just focus on the DRTR approach. The other place, Rhonda, where we do pay a lot of attention to protein is in the few of our patients that are taking GLP-1 agonists. So I've been a pretty public critic might be too strong a word, but I've certainly expressed my reservations about the ubiquitous use and the liberal use of GLP-1 agonists, especially in people, just trying to lose 10 pounds, right? Like, it's one thing if you're 100 pounds overweight and you've tried everything, by all means, the benefits clearly outweigh the risks. But, I got to get my beach body on for the wedding this summer. I'm going to lose 10 pounds. Let me fire up some semaglutide or tirzepatide. I think that's a net negative personally. And in those patients, not that we're giving it to those patients, but in any patient who's on a GLP-1 agonist, we feel it is so essential to hammer home protein, because those drugs are so effective at squashing appetite that, we've seen people who basically just want to drink alcohol when they're on it, and they'll lose weight like crazy because they're, not getting that many calories, but they're like, yeah, I just like wine.
Losing muscle.
Yeah.
Like,
I'm just losing muscle drinking wine.
Yeah.
So
yeah, I totally, like, I've got some acquaintances that are of that category where it's like, a stay-at-home mom wants to lose 10 pounds, has the means to get it and does it. And we haven't measured muscle, but I look at them and I'm like, you look like you're wasting your muscle is wasting. Like, if you're not eating, you're not taking in protein. So, that makes 100% sense. And it also, I don't know, maybe we'll talk about this when you come on my podcast, but like, I'd love to, cause you used to do a lot of fasting, and I know you've kind of, you don't do as much, at least of the long fast. And boy, I would love to get into that. You can decide whether we should do that next time, or we could talk a little bit now. But yeah, that was also like the biggest, there was another shift in my understanding of, fasting and time restricted eating. A lot of people use time restricted eating. They sort of practice it by skipping meals. And I don't know necessarily that's the way to do it, but people do that. It's just, what people do. And when you're skipping a meal, you're skipping your protein, right? And so you're not getting as much protein as you're basically becoming, losing muscle mass because you're not getting that important signal, especially if you're not doing resistance training, right? Then it's like kind of a disaster. And that was also something that I hadn't thought about a lot. And I know you've got a lot of experience in it, both personal and clinically. But
I'll share with you briefly how we think about that. So on the time restricted feeding part, we agree that the greatest drawback is that the patients get protein deficient. So time restricted feeding as a strategy for weight loss vis-a-vis caloric restriction is very effective with a small enough feeding window. So, a 16-8, you can eat your way into obesity with a 16-8. But once you start getting down to a 24 or a 22-2, basically just doing one meal a day, really getting restrictive, for the most part, you're going to lose weight. The problem is, by definition, you're not going to get enough protein in, because even if you managed to scarf down 1 gram per pound of body weight in a single meal, you wouldn't be able to utilize those amino acids. You kind of tap out at about 40 to 50 amino acids per meal. So if you sat there and had 160, you just flushed a bunch of them down the toilet, right? They're literally not coming down the toilet. They're coming out as vis-a-vis urine, right? Coming out the urea cycle. So the thing that we would counsel people on if they're going to use time restricted eating is they have to have protein snacks outside of their feeding window. So if they're going to say, look, I'm going to only have kind of a lunch at 2:00 and a dinner at 7:00, we'll say fine, but you still have to have 2 protein snacks outside of that. And that becomes challenging because those protein snacks can't really have much else in them. They have to be very low calorie otherwise. Otherwise, you're not really doing time restricted feeding. And of course, a lot of people get phosphorylated over this. They say, but oh my God, that's like outside of my feeding window. Will that impair autophagy? To which I argue, you're not getting any autophagy doing a single day time restricted feeding anyway. It doesn't matter. But if people are getting gut benefits from taking that time off, then yeah, they're going to miss out on those, because I just don't see how you can get the gut rest if you're trying to get those amino acids. And so you might have to really start to cycle those things. But yeah, long-winded answer to why I think fasting can really be at odds with the adequate maintenance of muscle. And as we get older, I think that I'm just entering my 6th decade. This is a very high priority for me.
Oh, really? Wow. You look great, Peter. I didn't. So you're in your
I'm
late
50.
50s?
No, 6th decade. So
yeah. So you're
just
you're
turned
50.
50.
Okay.
Yeah.
Gotcha. Yeah. Well, the
But
time
I love that you thought I was 60. That's awesome.
Well, you know who's
I'll take
Joe
that
I think Joe Rogan's entering that, and that's kind of what I was thinking. And he looks, I mean, like, that you can definitely see the people that like put in the work and work out and they do aerobic, they do strength training. Like, you look at them. There was like a study published on that too. Like a bunch of biological markers of aging and biomarkers of aging were measured. And then like there was like people looked at pictures and like ranked their age and their quote unquote biomarker biological age. So like according to all these biomarkers that basically say their chronological age may be older than their biological age. Well, they looked like their biological age, not their chronological age. So that's also important.
Although I sometimes feel like excess exercise can prematurely age you as well. I've certainly seen a lot of, and I don't know how much of that is the sun damage, because of course a lot of exercise is done outdoors, and of course sun can play a horrible role in that. I've taken up more of your time than I said I would, but I want to ask you kind of just one last thing. Is there any other, just sticking with this theme of things that you believe today that you didn't believe 3 or 4 years ago, or things that you believed 3 or 4 years ago that you don't believe today? Is there anything we haven't touched on? Because we've talked about some really good ones.
I think those are the really important ones off the top of my head. I definitely don't want to get into the whole COVID thing at this point, but my view has changed on things as that has progressed and changed as well. So I don't want to like not mention it, but yeah. So, but I think the most important things would be muscle mass, protein intake. Also, I like fasting, and I think the effects of time restricted eating on weight loss, specifically when you're looking at that outcome being attributed to caloric restriction. I think that is something that I wasn't always, buying into that, but it is still my opinion there are benefits to eating within your circadian rhythm. I mean, so like, eating late at night when you're making melatonin 2 to 3 hours before bed, you're basically inhibiting insulin secretion. And I don't like, there's data showing that you will, your glucose levels will be higher with the same exact macronutrient intake as if you eat it earlier. So there are benefits, circadian benefits. And I also think you mentioned the gut rest and like digestion, resting, like having that rest period, like your body can't, so DNA repair mechanisms, you mentioned autophagy, like those things happen when you're not digesting. So like, and that process like isn't happening. So you have to have like a rest period for repair processes to occur. And I don't know that I think autophagy is as good as the markers that we are sensitive assays that we have to measure it. And I don't know that it's settled with respect to how long. I personally think there's probably, even in between meals, there's some amount of autophagy. Autophagy is happening in us. It is. And it's not like we're not.
But is it clinically significant, right? Or more so than, say, exercise would induce?
Yeah, no, right. Exercise, nothing. Like the exercise is.
Like, that's my point. Like, how long would you need to fast to get the benefits? How long would you need to fast to get the benefits of an amazing workout? And my thinking is probably a long time. You might have to go a full day without food or a couple days without food to get the benefits of that. But you're right. I think without biomarkers, a lot of this stuff is very difficult to speculate on, because we can't really extrapolate from mice on this stuff. I think I don't, it's so non-linear that I don't think I could. And I've never heard anybody offer a very compelling argument either for what the quote unquote answer is.
I agree. We can't extrapolate for mice. And the way I view it, though, is more of a cumulative effect, where I'm thinking, I think it's better to just eat within a circadian window. And do I think that's going to have a cumulative effect on, metabolism? And yes, I think that it's better that I'm not eating, within a 15-hour window, like eating, which most people in the United States, they actually do eat, like they're eating from start to finish. Like a lot of people are eating like, within a 15-hour period. And so is that going to, like, if you're exercising, maybe it doesn't matter. Maybe you're right. Maybe they can eat within a 15-hour window. We don't really know, but I tend to think probably the circadian component like does play some role. But the question is it significant? Is it significant?
Yeah.
I am of the opinion it probably is cumulative.
If for no other reason, I think it just is on sleep, right? I mean, if
on sleep,
yeah, exactly.
Right.
Like if you just looked at the benefits of nighttime food restriction in terms of, as you pointed out, we're least insulin sensitive, and the negative impacts probably of thermogenesis and other things on sleep, that's probably the most compelling reason, even if nothing else mattered, if we couldn't measure it. But those are so abundantly clear, right? Like
mm-hmm.
that's as clear as how alcohol impairs sleep. A late night meal is a great way to destroy a good sleep.
Absolutely. Right. And I think most people have, it's anecdotally, like people realize that as well.
Yeah.
So yeah, I think we covered a lot of the things that have my perspective has shifted, as any scientist that's following data should. I mean, some people will argue, oh, you changed, how can I follow you? You're, changed your mind. It's like, well, like when new data comes out, like you have to reassess things. Like I reassess the supplements I'm taking. I mean, the supplements I take now versus 5 years ago, totally different.
Yeah.
Not all of them. There's some
right.
base things like that I like, vitamin D, omega-3, like those are like super important, I think. But, like you have to reassess things because new data comes out and, you might have a new understanding of things that we didn't know. We have new tools. We have, it's always getting better. So you have to kind of reassess things.
So Rhonda, for folks to follow you, obviously your podcast, Found My Fitness, great way to follow amazing content. Also, you put up a lot of content. I think Instagram is probably where you're putting up most of your content, right? Is that safe to say that if folks follow you on Instagram, that's where you're doing kind of really sort of thorough analyses and stuff like that? Would you recommend people also check out Twitter? Where should people be going to see your thinking on a frequent basis?
Well, it depends on what they like to consume. So if they like to consume like in-depth articles, we publish them on my website, foundmyfitness.com. We have like topic articles that we cover. Okay. We cover blood-brain barrier so we cover that more in depth. Some people like to read, they like to nerd out on that. So that would be the place for that. And then some people just like short little to the point, like what, and that is where, if they want like the sort of quick thing, like the Instagram would be, so Found My Fitness on Instagram and also Twitter as well. I do like a lot of, it's kind of like a short, I mean, you only get so much time on Twitter and, you can't go into in-depth nuance. And then I sort of have a love-hate relationship with Twitter, but it is kind of a fun place to also like, there's other scientists on there as well. And so, I'm on Twitter and Instagram, and
it's all the same handle, right? It's all
Found My Fitness. Yeah. Found My Fitness. And then the podcast as well, which is on Apple Podcasts.
Andrew Huberman and I were talking a little while ago, and we were sort of singing your praises as truly the OG health podcaster. When did you start? Was it 2014?
Yeah, that was when I started the podcast it was a weekend. I was doing my postdoc in Oakland, and I just started like Ron Krause down the hall, George Brooks. So I have a podcast with George Brooks on lactate. Like he was like my second
I don't
podcast.
think I've heard that. I need to go and listen to that one.
Yeah, we went all into the brain. I've been into that, like that whole podcast, like kind of shifted my thinking of like intensity of exercise and the importance of lactate. Because, I don't know if you know this, but like I trained, we probably shouldn't, like we already talked about where people should go find me, but like I was a mitochondrial metabolism, researcher in grad school, and so I was looking at the role of mitochondria in cancer. And so of course, like lactate, I mean, I was thinking about it in a completely different frame of mind.
Right. As opposed to, I mean, we now know it's such an important signaling molecule in addition to all those other things we talked about. It is We've had at least one guest suggest that George Brooks is deserving of a Nobel Prize.
I mean, I think, his lactate shuttle theory, it's called a theory, and I kind of hate that it's called that because people hear that and they're like, "Oh, it's a theory."
It's not proven,
But it's been proven. And in fact, there's like studies, like even like there were people looking at this like even before he proposed, like they were looking at lactate getting in the brain in response to physical activity, and it's like beta hydroxybutyrate. It's a signaling molecule. It activates BDNF like beta hydroxybutyrate. They go through the same transporter, monocarboxylate transporter, the MCT transporter, to get into the brain. And there's also those in mitochondria. But it's so funny because there's a lot of similarities, the effects on TBI. So, George Brooks has done some studies, USC, looking at, some of these victims of TBI and giving them lactate. And it's like improving whatever their Glasgow rating scores, whatever the things that they're looking at. But like, I think beta hydroxybutyrate, wasn't there some evidence, I think, with TBI? I remember Dom talking about that.
Dom has talked about this,
Yeah. So it's like, it's so interesting because also, with respect to like Alzheimer's disease, and like there's some really preliminary data that of course needs to be repeated, probably won't because you can't get funding, but like giving beta hydroxybutyrate, BHB, to people with Alzheimer's disease can like help improve, like there's some small clinical studies looking at improvement in like cognition. And, so lactate's like, I think similar. So there's a lot of overlap there. Now lactate you can make, beta hydroxybutyrate you make from exercise as well. You like push yourself into ketosis, right? So it'd be interesting, like there's, if there's synergy there, but like the trying to get the lactate and the beta hydroxybutyrate, the neurobiological effects of them, to me is like, it's so important. It's so interesting. And I kind of want more research in that area. And so I like talking about it because I know scientists listen to your podcast, researchers, physicians. So it's good to kind of like spread ideas. I mean, that's part of what the podcast does, right? Spreading ideas. It's not just like I'm communicating, the health ideas, and it's like scientists are listening to this and they're outside of their like, lens where they're only thinking about the thing that they research, and they hear this and it's like creativity, right? They start to go, oh, like, and I've seen that happen, like that, like scientists like doing experiments, based off of like listening to podcasts and stuff. And so I think it's really great. But thank you for the OG. Yeah, I did that, and it was a weekend thing for me, and I loved it. It took off. I love it so much. Like, I know you do as well. I mean, it's like, and by the way, phenomenal podcast. Like, it's funny. I don't listen to podcasts much at all, as I mentioned, but there's a few people that I trust to really be vigorous in their, research, and to be critical, to really like dive down and like get into the root of things. And you are like one of those people. And so like people will come to me, and say, oh, Peter or Tia said this. And I'm like, oh, okay, I should like, this is like, this is something I should consider. Or, oftentimes it'll be, oh yeah, Peter also said that. And so I'm like, oh, good. So I'm always like, okay, what is Peter thinking? What is Peter thinking? So it's nice to have you as a colleague, as a friend.
Likewise.
I'm glad we've reconnected. Thank you so much for inviting me on your podcast. Can't wait to speak with you again soon on your amazing book, which I can't wait to read and discuss as well in a couple of months.
Well, thank you very much, Rhonda. I can't wait to see you in person. Even though it's been pretty awesome to see you in video, your setup is, as I said, exceptional. So we've treated the people who are watching this to a world-class view of what a home podcast setup can look like.
Amazing. Thanks, Peter.
All right. See you, Rhonda.